Within 5 minutes, we are waiting for some of Okay, I now this is really academic session. I want the is lecture providing is for this second session. I request for the organizers we will to decisions now. Along with the Shastra, we will develop decisions now. And what the professors are going to session. Introduction of communication to assistance. For a nice introduction. Today, before starting the lecture, I would like to thank Professor Arvind Kumar, Principal SD College of Pharmacy, Sri Neeraj Kumar, the member of the trust. I think he's also one of the trustees in the SD group of colleges. So I thank Dr. Vaisali who contacted me for delivering a talk in this conference, international conference, which is organized by SD College of Pharmacy second time. And I'm fortunate to be here as I was also present during the first time in 2024. So thank you SD Group of Pharmacy for inviting me here. Before starting my talk, बच्चों लास्ट स्टेज में जो बैठे हो कैन यू कम फॉरवर्ड आगे आ सकते हो आप एम आई ऑडिबल because I am giving the responsibility to make the session more interactive, so I have to ask one or two questions from the students. How many of you heard the word drug repurposing? Students, have you heard this term, drug repurposing? Any student who has heard this term, drug repurposing? Okay, but I will give you an example which you will understand that you already know this term, drug repurposing. So, I think, have you heard about Disprin tablet? Okay, very good. This side, what is the use of Disprin? For which you use the Disprin? Headache. Okay, and what is the drug? What is the drug inside it? In Disprin, what is the name of the drug? Aspirin. Okay, so Disprin was used as an analgesic. Now also it is used. But nowadays, aspirin, another brand, Ecosprin. It comes with the name Ecosprin, and it is very much recommended to the cardiac patients. For what use? For which use? Blood thinner. anticoagulant property. It means the drug was already available in the market and it is repurposed. It is searched for another use for the benefit of the community, for the benefit of the patient. So this is all about the repurposing. So such process, such techniques, we call it drug repurposing. So the title of this conference, Translational Research for Sustainable Drug Discovery and Environmental Health. First slide. Can you? So, what is translational research? The process of converting basic scientific research into the clinical applications. With whatever research we carried out at college level, university level, basic research. Okay. It is of no use if it is not ultimately get converted into for the clinical applications. Okay. So such translation, such transition, it is known as translational research. Okay. It links the research laboratory practice, public health, like we conduct clinical trials. It accelerates the development of therapies, diagnostics. Another alternative in the drug development strategy is exploration of drugs that have already been approved for the treatment of disease and whose target have already been discovered. As an example of this aspirin. So translational research, it helps to bridge the laboratory research to real world health applications, which we have to convert any scientific research into the clinical application. Next. Here. Another term, the drug discovery. You know, 15 to 20 years back, how much time you can expect a new drug molecule to come to the market? Any student, faculties, how much time a molecule takes to get recognized as a drug? Okay, as you can see in the picture, it takes around 15 years, 15 to 20 years. So it's a very lengthy process, very costly affairs. Out of 10,000 molecules, which when the research labs, the different pharmaceutical companies, they start working with different molecules. Out of 10, you can say we get only one molecule which can be translated into a drug molecule. So because it takes a lot of time, that's why it's very difficult to discover new molecule, but whatever drug we have, whatever natural products we have, we can search them, we can carry out our research on them to find their use in the other diseases for which no drug is available in the market. Like what happens in COVID? Can you name any medicine, any drug which was initially tried for the treatment in COVID? Any drug? Remdesivir, yes. Okay. So when this drug was tried, okay, the prices of this drug, they got shoot up. Okay, earlier this drug is a antiviral drug. Okay, so they, the scientists or the different researchers, they thought it may also work against the COVID. Another drug, chloroquine. Have you heard about chloroquine? Chloroquine, a lot of chloroquine was exported from India to several countries because India is a big manufacturer of chloroquine. So, drug discovery, if we say, it is a process of identifying chemical entities, new molecules that have the potential to become therapeutic agents. There are chemistry people, they synthesize a number of chemical compounds, number of derivatives, but out of those derivatives, hardly one or two becomes successful in becoming a drug molecule. So, there are three stages in the discovery: lead identification, preclinical testing in the animals, then clinical testing. Means in drug discovery, first thing is on which drug, on which disease you are working. First, you have to identify the disease. Say diabetes. Okay. First disease, then you have to identify what are the receptors. What are the receptors? What are the receptor proteins? Okay. Then you have a different compounds which we call them ligands. So we have to. So one is ligand, another is target. Clear? A receptor may have different proteins, but which protein is most suitable for a ligand? That we study in this drug discovery. So, in order to avoid such a long-term process of conventional drug discovery, reverse engineering process is also gaining importance. Another alternative in drug discovery strategy, drug development strategy is exploration of drugs that have already been approved for the treatment of diseases. Okay, so this is drug repurposing, what I say. So next slide. Yes. As you can see, there are in this flow chart there are two charts, one above and second is below one. This. So most of the discoveries they came to this world by serendipity. Serendipity means discovery by chance. Discovery by chance. So, in one type of research where lead compound is there, okay, it is tried against a particular target, okay, then out of several ligands we get a drug candidate, one active molecules, okay, that molecules is first tried in the animal studies, preclinical stage. Once it clears the preclinical stage, then we try it in the humans, clinical trials. Okay. Once the drug molecule it passes the clinical trials, then it is registered as a drug molecule and permission is granted to a particular pharma company, okay, to be marketed by for the treatment of disease. But in repurposing, this is traditional drug discovery. But in repurposing, we know the ligand, we know its mechanism, we know its receptors, its disease receptors. So we have to try it in different receptors of different disease. Okay. So we have already clinically approved drug, like in case of aspirin, it is already clinically approved. So another, we have to select the targets of new disease. We have to validate them. Then clinical
clinical trials, registration, market. So, drug repurposing, it is a cost-effective method for the pharmaceutical industry and beneficial in situations like COVID. Okay, this technique works. Repurposing. So, if you see what is drug repurposing, it is retasking, drug reprofiling, drug rescuing, drug recycling, drug redirecting, and therapeutic switching. Therapeutic switching means from one disease, the drug is switched to next disease, another disease. Okay, so drug reprofiling, retasking, recycling, redirecting, all are same terms which are used for drug repurposing. So, what actually is repurposing? It is a process of identification of new pharmacological indications. New pharmacological indications. Identification of new pharmacological indications from the old or existing drugs, old drugs, failed drugs, the drugs which failed in clinical trials, say phase 1, phase 2. Okay, but it raised the clinical trials, but could not come to the market due to one or other reasons. Investigational drug, already marketed drug, FDA approved drug, or prodrugs. Means if any of such a drug which lies in any of these categories, okay, we have to identify such drug which can be used for the treatment of another disease. This is drug repurposing. I'm again telling, like in case of aspirin was the drug, non-drug, okay, existing drug, but it was identified for new pharmacological indications. That is for to prevent the coagulation of blood. That's why it is recommended to the patients, okay, which have blood clotting problem. So, and applications of newly developed drugs to the treatment of diseases. In drug repurposing, you have to identify a drug molecule. That drug molecule, it may be existing molecule, or it may be old molecule, or it may be a molecule which got failed in the clinical trials, or it is in the market, or FDA approved, or prodrug, or under investigation. So, application of the newly developed drugs to the treatment of diseases other than the drug original or intended therapeutic use. Means previous use of the drug, the new use of the drug should be different from the previous use, previous indications. Aspirin, earlier it was indicated as a analgesic, but now it is indicated as an anticoagulant. So this is repurposing. The use of to find out the use of the same drug for another disease. So, it is an act of taking a drug intended to treat one patient population and demonstrating its efficacy in the treatment of a completely different group of patients. That is also drug repurposing. Next. So, as in the previous slide, we went through different terms: drug repositioning, drug repositioning, drug reprofiling, drug reformulating, drug rediscovery. So, drug repurposing, finding new uses outside the scope of the original medical indication for the existing drug, or developing new indications for the existing drugs or biologics. Means किसी ड्रग का जो यूज है किसी पर्टिकुलर डिजीज में, उसके अलावा किसी दूसरी डिजीज के अंदर कोई दूसरा उसका एप्लीकेशन मिलता है या प्रूव होता है, दैट इज ड्रग रिपोजिशनिंग. वी आल्सो से repositioning from one position to another position. Same. Repurposing, identification, developing and commercializing new usage for existing or patent drugs. Means existing drugs are there or a patent drug is there, but if we can identify, develop and commercialize new use, it is repurposing, reprofiling. Same thing, reducing the risk and associated with the drug developments, with the advantage that drug has already undergone preclinical and clinical testing. Some drugs which have crossed the preclinical stage, it entered the clinical stage, but due to some reason it could not clear the phase 4. It reduces a loss of lot of economic burden on the pharmaceutical company. So this is reprofiling, reformulating. As you can see in formulations, drug is there, but earlier a drug was formulated in the form of a tablet. Later on it was formulated as a microsphere. Now it is formulated as a nanoparticles. Okay, mean drug is there, but it's different formulations are different. So finding ways to modify the formulation to allow a drug to enter a new market. Drug rediscovery. Drug is there, but we are discovering its new use. Investigating new uses for currently prescribed drugs. Next, please. Yes. So these are the approaches and the resources by which which can be applied in the drug repurposing or repositioning. Say experimental based repurposing, clinical based repurposing, computational based repurposing. You can see these days a lot of computational modeling, docking, simulation studies are carried out to screen number of already existing compounds which are drugs or old drugs. So we can use the computational approaches. We can use clinical based repurposing. Okay. So like binding affinity approach, phenotypic screening, and this in clinical based repurposing, knowledge based approach, target molecular docking based approach, pathway mapping based approach, target mechanism based approach, machine learning, network models, genetic association based approach, signature based approach, text mining, and semantic approach. Means a lot of AI/ML applications are available with which we can screen the drug for its to find its use in another disease. Next please. Another model, de novo drug discovery route. It is now struggling de novo drug discovery, leading to the search for a new model to accelerate delivery of treatment to the clinics. With the purpose of translational research is whatever research we are carrying out, whatever compound we are synthesizing, either at the college level or university level, ultimately it has to come to the society for some health benefit. Otherwise, it is useless. So, drug repurposing is one of such possible models in case of the drug discovery. Next, it's possible targets. So, existing generic drugs, whatever generic drugs are available in the market, so that are available in the market for one or more existing illnesses. With the existing drugs which are available in the market and prescribed for some indications. They can be taken in the drug repurposing. Several compound means several compounds which were synthesized, which cleared the preclinical studies, they entered the clinical trials, but they could not cross the phase 3 or phase 4 due to one or another problems. Okay, so in that case also, means what we do, we kept them in the store, sell the compound, means that they have potential, but due to some reason they could not be converted into drug molecules. So several pharmaceutical companies, they have self compound, they prove to be safe in humans, but did not prove efficacious in their original targeted illness. Another type, the current branded compounds. These are available in the market, but remain protected by intellectual property. Say, certain drugs which have certain patent rights, so other companies, they have to wait till the expiry of their patent period, but they can carry out research. Next, the challenges and opportunities in the drug repurposing, because in this case we have to optimize the process for inclusion and exclusion criteria in the selection of target population. The drug, which type of target population we have to include, which type of population we have to exclude, okay, we have to optimize it. Second, achievement within timeline. Okay, whenever we carry out research in the repurposing, so we cannot say that within 1 year, within 6 months, or we will be able to give you the final results. Requirement for robust evidences, lack of funding to commercialize the product. Most of the companies, they are struggling with this economic crisis. Clinical trial feasibility for rare diseases because of the strict ethical guidelines of the governments. Technical challenges for to repurposing the drugs. Novelty. One of the important criteria in drug repurposing, there should be some novelty. And what is this novelty? That the drug should be indicated for a new disease. The drug should be prove its efficacy in some different pharmacological indications. Next, please. So, have you heard about the name of this digitoxin? Digitoxin, digitalis plant. Most of the students, they have studied in the B.Pharm. Okay. Earlier, digitoxin, it was well known for, it was intended for cardiac arrhythmias and heart failure. But now, by repurposing, it has been indicated for ovarian cancer. The drug is same. Okay, now it is indicated for another illness. So this is repurposing. Bupropion, a drug well known for this depression, antidepressant drug, but now it is used in the smoking cessation. If lornithine, antiparasitic application, earlier not approved for antiparasitic application, but by repurposing, it is used for this for the treatment pneumocystis, carry pneumonia in the AIDS. Next, finasteride, benign prostatic hyperplasia type of cancer. Okay, but now it is also proved its use in preventing the hair loss. Galantamine earlier uh, indicated for polio paralysis anesthesia. Now it is indicated for this Alzheimer disease. Drug is same, but use is different. Next, celecoxib, earlier prescribed for osteoarthritis as anti-arthritic drug or in rheumatoid arthritis, but now it is used in adenomatous polyposis. Infliximab, earlier recommended for Crohn's disease, but now it found application in juvenile rheumatoid arthritis. New application. That is saying new application. Heparin. Earlier heparin, heparin as an anticoagulant drug. Okay, but now it is indicated as cyst in preventing the cystic fibrosis. Ketoconazole, antifungal, we all know. Okay, so recommended for controlling the systemic fungal infections. Now it is also used in indicated for nephrotoxicity. Albuterol, it is used to control the bronchospasm, exercise-induced bronchospasm, but now it is also used for the prevention of paralysis due to spinal cord injury. Leflunomide, earlier indicated for arthritis, now it is indicated in myeloma type of cancer. Next, nitroprusside, earlier indicated for acute congestive heart failure. Now it is used when cerebral vasospasm. Phentolamine, earlier used to prevent this dermal necrosis. Now it is prescribed for autistic patients. Autism. Propinerol, earlier used as an antihypertensive drug, now used to control the restless leg syndrome. Next, sodium thiosulfate, earlier used for cyanide poisoning, now used for new, after drug repurposing, the new use platinum-induced ototoxicity in pediatric patients. Topiramate, anti-epileptic drug, but now it is also found its application as anti-obesity and IBD. Next, Chidorubin, as we know, earlier this drug was discovered for its fact as anti-cancer drug, but now later on anti-HIV. Next, artemisole, earlier it is used to prevent the allergies. Now it is also find its application in malaria. Sildenafil, earlier already available, mostly used this drug for erectile dysfunction, but now it is also used for another problem, African sleeping sickness disease. Metformin, antiductive drug. Now it is also recommended for the treatment of polycystic ovarian syndrome, a problem in most of the people. Minoxidil, as a antihypertensive drug, it was earlier approved as an hypertensive drug, but later on it also approved for promoting the hair growth. Now, so I gave you several examples. So, the purpose of drug, the advantages and the purpose of drug repurposing. It saves the time, it reduces the cost several times, as compared to the conventional drug discovery. So it is an effective strategy to find new indication for existing drugs and is highly efficient, low cost and riskless, because most of the toxicity study to the already known drug or existing drug that have also already been completed or tried by in the trials. There are only 5 steps in drug repurposing: compound identification, ligand identification, acquisition, development, FDA post-marketing, market surveillance or safety monitoring. Means at this stage we have to see whether the drug is showing any adverse effect at that dose. However, most of the drug which are discovered in the, after repurposing, they are riskless. Still, we take the feedback once it is launched in the market. Due to fast growth of bioinformatics knowledge and biological big data, drug repurposing decreases the time and cost of drug development process significantly. Definitely. Next. So, thank you very much for hearing me and listening me. I think, dear students, I think now the meaning of the drug repurposing it should have been clear in your mind. Means you should have understood the meaning. What is the repurposing? So, any questions related to repurposing from the students?
Session is now open for question and answers. Any student, any participant, if there is any query or discussion he or she want to make, she can. Anyone from the participants?
अच्छा,
Yes.
कोई बच्चा एक एग्जांपल और बता सकता है? एस्पिरिन के अलावा कोई दूसरा एग्जांपल repurposing का? Yes, minoxidil. Students. कौन सा? डिजॉक्सिन. Yes. दूसरा कोई? कोविड में क्या हुआ था? कोविड में बहुत सारे मेडिसिन ट्राई किए गए थे ना. क्लोरोक्विन किया था, रेमडेसिविर किया था. वो क्या था? वो repurposing था. Means drug was already available in the market. We knew the drug, but we want to check whether it can act against the new virus which have created the pandemic, which was responsible for the pandemic. So, any other questions? Students. So, the BPharm students who want to pursue further studies in MPharm or PhD, they can work on this drug repurposing. You can design your project on the repurposing, because you know the drug, you know the current indications, you know its targets, you know its toxicity. Now you have to see whether in its mechanism of action, its pathway, whether any other enzymes, any other protein is involved to which this drug can affect or not, it can impact or not. So, by molecular modeling, simulation, you can carry out at initial level. Okay. So, thank you from my side. So, I am once again thankful to Professor Arvind Kumar ji, Principal SD College of Pharmacy, for giving me the opportunity, for inviting me here. Dr. Vaishali ji, inviting me here. So, thank you very much.
Thank you so much, Dr. Dinesh Kumar sir, for your very impactful, informative insights on drug repurposing. Now I request Dr. Arvind Kumar, Director, SD College of Pharmacy, along with Dr. Saurabh Sharma sir, Director, Vivek University Pharmacy Department, along with Dr. Vitesh sir, RVIT College of Pharmacy, Bijnor, to felicitate our Honorable Speaker of today's session 2, Dr. Dinesh Kumar, former Head Department of Pharmaceutical Sciences, Central University of Haryana, Mahendragarh. Alongside, I request oral presentation participants of oral and poster presentation, please move towards Pharmacy Department. Participants of oral and poster presentation, please move towards Pharmacy Department. I request our dignitaries to felicitate our session chair and co-chair of this session, Dr. Rizwan Ahmed sir, Dr. Vishal from Vivek University, Bijnor, along with Dr. Ajit Sanskar, Professor Sanskar College of Pharmacy. Dr. Vishal, Dr. Ajit. Yes, sir. I request Dr. Govind sir also to please join us on the stage, please, for the felicitation ceremony. Please, sir. After this, I request and would like to felicitate our prestigious advisory board members. In the same queue, I request Mr. Lavi Kumar, Assistant Professor from Krishna College, Bijnor, to come up. He is a prestigious member of our advisory board from Krishna College, Bijnor. Mr. Lavi Kumar, please, sir.
I request Dr. Popin Kumar sir to please come on the stage. Dr. Popin sir.
Ms. Rupa Nirwal, Assistant Professor, Krishna College of Pharmacy, Bijnor. Ms. Rupa Nirwal. Is she available, Ms. Rupa Nirwal? Ms. Vijeta Sharma. She is a senior resource scholar in Federal Department of Cell Culture. Senior resource person, Ms. Vijeta Sharma. She is a prestigious advisory board member of this 2 days CSTUB sponsored international conference BioStart 2.0. Next, I request Ms. Lakshmi Saini, Assistant Professor for Pharmac College of Pharmacy. Ms. Lakshmi Saini, Assistant Professor, Pharmac College of Pharmacy, UP. I request Honorable Dr. Vijay Kumar sir, Professor, Mahavir College of Pharmacy, Meerut. He is a prestigious advisory board member of this 2 International Conference BioStart 2.0 sponsored by UPCST. Mr. Dr. Vijay Kumar, Professor, Mahavir College of Pharmacy. After this, I like to thank and also want to welcome our sponsors of this 2 days conference, Escoven Innovation from strong motion, I would like to invite Ms. Pratibha Maurya ma'am along with Dr. Ajit. Ms. Pratibha Maurya ma'am from Escorville Innovations Private Limited along with Dr. Ajit Maurya. And the same I request Dr. Okay. Our next sponsor from Volga AI, Mr. Chirag Work. Mr. Chirag Work from Volga AI. I request Karan Bhagwat to be ready for the poster presentation after this session. Thank you so much, all the dignitaries, members. I request you to take your respective places and seats. And I request Dr. Ajit sir to, he will be sharing very insightful version of this corporate innovation private limited, a venture established by him and Ms. Pratibha Maurya. Ajit sir.
हेलो टू एवरीवन। अभी आपने एक नाम सुना एस्कॉर्विन इनोवेशंस। पिछले कुछ 7-8-10 सालों से मैं एस्कॉर्विन इनोवेशंस के दूसरे फॉर्म के साथ एज एडवाइजर जुड़ा था। तो कई सालों से हम लोग इस पे काम कर रहे हैं। अभी जस्ट 10 मिनट पहले डॉक्टर दिनेश सर ने एक बहुत ही बढ़िया कॉन्सेप्ट आप लोगों को समझाया था, ड्रग रिपर्पसिंग का। तो ड्रग रिपर्पसिंग का कॉन्सेप्ट बहुत ही बेहतरीन है, बट उसको फलीभूत कैसे किया जाए, उसको मटेरियलाइज कैसे किया जाए, इस पूरे कॉन्सेप्ट को डिजाइन करते हुए हमने एक सॉफ्टवेयर का डेवलपमेंट किया है। जिस तरीके से आप इन विवो केसेस में किसी भी ड्रग को चेक करते हैं, सेम वैसे ही बिना बॉडी में डाले आप किसी भी मेडिसिन को उसके मशीन के फॉर्मेट में एक वैल्यू आपको मिलेगी जो कि बिल्कुल इन सिलिको को रिजेंबल करती हुई होगी। तो अभी सर ने आप लोगों से कहा कि आप लोग इसमें बहुत ही प्रोजेक्ट कर सकते हैं, पर प्रोजेक्ट करने के लिए आपके पास कुछ होना तो चाहिए। आपके पास क्या टूल्स हैं, क्या टेक्निक्स हैं, उसको रिजेंबल करता है। जैसे इन विवो कॉन्सेप्ट है, वैसे ही वी आर लॉन्चिंग इन मैक, जो कि मशीन जनरेटेड वैल्यू है और इन विवो वैल्यूज को बहुत रिजेंबल करती है। अब आप कहेंगे कि रिपर्पसिंग के लिए बहुत से ऐसे ऑलरेडी सॉफ्टवेयर्स अवेलेबल हैं जो लोग यूज भी कर रहे हैं और नो डाउट उनके बहुत अच्छे रिजल्ट्स हैं। लेकिन क्या आपने कभी ये सोचा है कि अभी तक जितने भी सॉफ्टवेयर्स अवेलेबल हैं ड्रग रिपर्पसिंग के लिए, उसमें आपको केमिकल स्ट्रक्चर स्पेसिफिक काम करना पड़ता है। मीन्स कि सपोज कि अगर आप फेनिटोइन स्ट्रक्चर की आप बात कर रहे हैं, तो आपको वैसा ही फेनिटोइन का डेरिवेटिव लेना पड़ेगा सेम टाइप की एक्टिविटी के लिए, या अगर आप रिपर्पस करना चाहते हैं तो 100% उस केमिकल कंपाउंड का स्ट्रक्चर डिफरेंट होगा। तो आप कैसे डिसाइड करोगे, कैसे सोचोगे कि मैं किसी पर्टिकुलर ड्रग को रिपर्पस कर सकता हूँ किसी पर्टिकुलर ड्रग के साथ? तो उस ड्रग के साथ रिपर्पस करने के लिए हमने इनमैक लॉन्च किया हुआ है। और मैं आप लोगों के लिए बता दूं कि इस कॉन्फ्रेंस में जितने लोग भी रजिस्टर्ड हैं, सारे रजिस्टर्ड कैंडिडेट्स को एस्कॉर्बिन इनोवेशंस अपने तरफ से एक ऑफर डिक्लेयर कर रहा है। वो आप लोग के पास आपके किट में भी है, जिसमें आपको 50% ऑफ मिलेगा, और अगर सर ऑर्डर करेंगे तो मैं आपको 100% फ्री मैक सॉफ्टवेयर दूंगा, जो ड्रग रिपर्पसिंग के लिए अदर सॉफ्टवेयर से बेटर रिजल्ट्स देता है। इसकी मैं 100% गारंटी ले रहा हूँ। क्यों? क्योंकि आज केमिकल स्ट्रक्चर स्पेसिफिक काम करते थे, अब आपको केमिकल स्ट्रक्चर स्पेसिफिक होने की जरूरत बिल्कुल भी नहीं। एक सिस्टम बायोलॉजी के बेस पे डेवलप किया गया सॉफ्टवेयर है, जिसमें जिस तरीके से आपकी बॉडी बिना केमिकल स्ट्रक्चर के ही केमिकल मॉलिक्यूल्स के साथ इंटरेक्ट करती है, वैसे ही ये सॉफ्टवेयर भी बिना केमिकल स्ट्रक्चर को बताए आप कोई भी केमिकल स्ट्रक्चर वहां पर इंट्रोड्यूस कीजिए, और ड्रग रिपर्पसिंग के बेस पे वो अननोन स्टैंडर्ड कंपाउंड के साथ आपको कंपेयर करके देगा। दैट्स ऑल। और बहुत सारे इन सिल्को कंप्यूटेशनल सर्विसेज भी एस्कॉर्बिन इनोवेशंस प्रोवाइड करता है। यहाँ के सारे रजिस्टर्ड स्टूडेंट्स के लिए हर सर्विस पे 50% ऑफ है अगले 1 साल तक। ये डायरेक्टर सर डॉक्टर अरविंद सर के वजह से पॉसिबल हुआ है, उन्होंने ये मौका दिया। तो अगले 1 साल तक आप में से कोई भी, चाहे वो टीचर हो, प्रोफेसर हो, स्टूडेंट हो, कोई भी अगर यूज करना चाहता है तो वो यहाँ पर रजिस्टर्ड है, तो अगले 1 साल तक वो हमारी सर्विसेज 50% डिस्काउंट पे अवेल कर सकता है। थैंक यू सो मच फॉर गिविंग द अपॉर्चुनिटी। थैंक यू सो मच।
Thank you, Dr. Rajit. And I have used your softwares and your services, and I know it's very wonderful and also very impactful. So, anyone if want these services can go to his website. And moving next, I request Mr. Chirag was from Volga AI, and he also sharing some of the inputs of his company and product and service. Please, sir.
Hello everyone, good afternoon. So, Volga AI के बारे में मैं बेसिक लेवल पे स्टूडेंट्स को इन्फॉर्म कर देता हूँ। हमने एक ट्रेंड नोटिस किया ऑलमोस्ट वे बैक 2 years बैक, एंड हमने देखा कि कॉलेजेस में प्लेसमेंट्स के लिए फॉर्म्स तो आ जाती हैं, बट स्टूडेंट्स उस इंटरव्यू की उस फॉर्म के लिए प्रिपेयर अच्छे से नहीं कर पाते। ठीक है, तो इसी चीज को हमने देखते हुए अपना टूल बनाया, जिसमें कि आपको सिर्फ जेडी डालना है, बीइंग अ स्टूडेंट, जैसा भी जेडी है, इररिस्पेक्टिव ऑफ डोमेन, टेक्निकल है, नॉन टेक्निकल है, मैनेजमेंट है, फार्मा से रिलेटेड है, इररिस्पेक्टिव ऑफ द डोमेन एंड जेडी व्हाटएवर इट इज, आपको जाके उधर जैसे अपना जेडी अपलोड करना है। आपको एक रोबोट आएगा आपके व्हाट्सएप पे क्रॉल करते हुए, और वो आपसे कन्वर्सेशन करेगा और आपको एक लिंक देगा। उस लिंक पे जाके आपको अपना इंटरव्यू देना है। अगेन वो इंटरव्यू आपका एक एआई मॉक इंटरव्यू, जिसमें हमारा एआई एजेंट आपका इंटरव्यू लेगा, और आपको पूरे इंटरव्यू खत्म होने के बाद ये बताएगा कि आप में कहां कमियां हैं जेडी के बेसिस पे, कि आपको इस चीज के लिए तैयारी करनी है, आपकी कम्युनिकेशन स्किल्स वीक है, आप नॉलेज डोमेन में वीक हो, आपकी आई टू आई कांटेक्ट मिसिंग है, आपके वोकल स्किल्स अच्छे नहीं है, आपको आईडिया नहीं है कि एग्जैक्टली कैसे करना है, आप बता रहे हैं, आपकी प्रणाली बात देगा और आपको ग्रूम करेगा एक रियल टाइम। ये टूल हमने बेसिकली स्टूडेंट्स के लिए, और ये टूल बिल्कुल फ्री है। कोई भी स्टूडेंट हमारे प्लेटफॉर्म पे आके इसको यूज कर सकता है फ्री ऑफ कॉस्ट। स्पेसिफिकली फॉर एससी कॉलेज हमने इसको बिल्कुल फ्री कर दिया है। कोई भी स्टूडेंट आए इसको यूज करे और अपने आप को इम्प्रूव करे। थैंक्स अ लॉट एवरीवन।
Thank you, Mr. Chirag Burks. And now I request okay, right. So we have between us Vagwa, which is a PhD scholar from MTU Rohtak.
One of the participants of our today's oral presentation. Sir, please. Karan Vagwa.
Good afternoon everyone. Myself Karan Vagwa, PhD scholar in the Department of Pharmaceutical Sciences, Maharashtra University. So currently I am just presenting one of my research work based upon the title Establishment of Novel Chromatic Induced adult zebrafish model of one of the novel chemical induced model which is an alternative to hypoxia. This work is basically carried out in the zebrafish research facility at Department of Pharmaceutical Sciences, Maharashtra University under the kind supervision of Professor Govind. Next slide please. Cerebral ischemia is one of the largest morbidity causing diseases because it is the fourth leading cause of death worldwide. As per the epidemiology statistical data of global burden of disease, which stated that the ischemia is largely responsible for the disease-associated lifestyles. The basic cerebral ischemia, we can say, is the part of cerebral stroke which is accountable for the 80% of the ischemic events. It is pathophysiological. It is majorly initiated due to the loss of blood flow to the brain tissue due to the blockage or reduction in any cerebral circulation. होता क्या है बेसिकली कि किसी थ्रोम्बस फॉर्मेशन की वजह से या बॉडी में एम्बोलिज्म की वजह से जो हमारे ब्रेन की वेसल्स होती है, जिनको स्पेसिफिकली बोलेंगे लार्ज ब्लड वेसल्स या कुछ हमारी स्मॉल ब्लड वेसल्स होती है, वो ऑब्लूड हो जाती है, जिनकी वजह से क्या होता है कि हमारी बॉडी में काफी सारे पैथोलॉजिकल चेंजेस आते हैं एंड दीज चेंजेस रिजल्ट्स इन दी कंडीशंस ऑफ मॉर्बिडिटी, जिसमें क्या पेशेंट चल नहीं पाता, पैरालिसिस एंड कंडीशंस की एक तरफ से उसकी मूवमेंट नहीं हो पाती. ये प्राइमरीली होते हैं एक्साइटोटॉक्सिसिटी जो उनके डिजीज में होते हैं. सो वी आर नॉट कमिंग अबाउट द पैथोफिजियोलॉजी. नाउ वी कन्वर्ट टू द नेक्स्ट पार्ट व्हिच इज व्हाई वी चूज द zebrafish as a model, because many of the researches are preclinical researches are involved with rodent models who largely use large animals, but for we choose a specific zebrafish model which is one of the novel model itself. Why we choose zebrafish as a model? Specifically we are using adult zebrafish, not larva zebrafish. Okay? So why we choose zebrafish as a specifically adult zebrafish? The very one feature of the adult zebrafish is that these are cost effective and ethical comparative to the rodents. जो अगर हम बात करें काफी एक्सपेंसिव आते हैं, IAC की अप्रूवल लेनी पड़ती है, हर चीज करनी पड़ती है, बट कंपैरेटिवली zebrafish में इतना नहीं है. अगर हम बात करें कि एक गाइड जितने के अंदर आता है तो हम कम से कम 5 से 10 zebrafish ला सकते हैं, बट एक कंडीशन और साथ इंक्लूड होती है कि IAC की अप्रूवल zebrafish में चाहिए कंपैरेटिव टू rodents में कि ज़्यादा IAC की अप्रूवल के लिए बहुत ज़्यादा डिटेल इन्फॉर्मेशन प्रोवाइड करनी होती है, बट ऐसा zebrafish के अंदर नहीं है. सेकेंड पोजीशन की ब्रेन स्ट्रक्चर, बात करें कि जो उनका ब्रेन स्ट्रक्चर है इट इज कंपैरेटिव सिमिलर टू द ह्यूमन. हम बात करते हैं दे हैव अ हाईली कंजर्व न्यूरोट्रांसमीटर सिस्टम जस्ट लाइक ह्यूमन. दे आर include serotonin, dopamine, glutamate, which are largely involved in the cerebral ischemia as well as high throughput screening कि हम एक साथ बहुत सारी ड्रग्स को मल्टीपल अपना एक साथ काम कर सकते हैं. देन वी हैव मॉलिक्यूलर लेवल्स की जो बात करें सेम जिस तरह से रैट के अंदर 80% ऑफ हमारे क्या कहते हैं जीनोमिक सिमिलैरिटी है या 80% हम बात करें कि जेनेटिक होमोलॉगस है, सेम zebrafish का जो क्या है 70% क्या है जेनेटिक होमोलॉगी सिमिलर टू दैट ऑफ द ह्यूमन. The large imaging, because the imaging of brain is quite difficult. अगर हम rodents के अंदर करते हैं तो हमें भाई नॉन इन्वेजिव टेक्निक्स की रिक्वायरमेंट होती है, बट इन द केस ऑफ zebrafish क्वाइट इजी. हम क्या करते हैं डेक्टिव फ्लोरोसेंट डाइस का यूज करते हैं और डायरेक्ट इमेजिंग करते हैं हमारी zebrafish के अंदर. नेक्स्ट स्लाइड प्लीज. सो व्हाई वी चूज zebrafish, सेम हम पहले डिस्कस कर रहे थे, इट इज अ हाईली कंजर्व्ड न्यूरोकेमिकल सिस्टम्स कि उसके अंदर गाबा का लेवल सेरोटोनिन, डोपामिन कंपेयर टू ह्यूमन के अंदर लार्ज प्रेजेंट होते हैं. सेकंड, दे कैन इजीली रिप्रोड्यूस ऑक्सीडेटिव स्ट्रेस, दे कैन इजीली प्रोड्यूस माइटोकॉन्ड्रियल डैमेज, दे आर लार्जली इन्वॉल्वड इन द न्यूरोइन्फ्लेमेटरी कैस्केड. सो दे मिमिक्स अ सिमिलर काइंड ऑफ पैथोफिजियोलॉजी दैट वी ऑब्जर्व इन ह्यूमन बॉडी. द थर्ड, वी कैन इजीली परफॉर्म बिहेवियरल असेस बिकॉज़ दे आर लार्जली wide number of behavioral assays in adult zebrafish that are widely used, including Roman time to determine their anxiety-like behavior. We have light and dark behavior to determine their scototaxic behavior. We will also use open field to determine their anxiety-like behavior and locomotion. And for that, cognition and memory aspects, we will use TMS and object recognition. So similarly, these zebrafish models are highly reproducible for the high frequency screening and mechanistic insight. So based upon certain parameters, we selected why should we use zebrafish as a model
तो बत्ती लाली घर की
Why
भाई
we choose specifically a bonnet ring? Because earlier there are various other methods for the induction of
मुझे दे हेलो हेलो
specifically तो पूरी large quantity of pathophysiological mimics जो है zebrafish के अंदर प्रोड्यूस करे. Why we choose chromatography? Because it has a certain pathological pathways that are relevant to ischemic stroke. Firstly, it is largely causing the central vascular endothelial injury and dysfunction. हम बात करते हैं कि जो हमारी बॉडी के अंदर vascular endothelial growth factors होते हैं, उनके जो एक्सप्रेशन को है chromatography बहुत ज़्यादा अमाउंट में कम करते हैं. Secondly, it plays a large role in the formation of thrombosis. जितने भी क्लिनिकल केसेस हमने देखे उनके अंदर ज्यादातर ये पाया गया कि क्रोमेटिन के एक्सपोजर से ही क्या हुआ है कि थ्रोम्बोसिस लाइक कंडीशन प्रोड्यूस हुई. जब थ्रोम्बोसिस बना वो ब्रेन के अंदर गया और जो स्मॉल वेसल्स ऑक्लूजन है उसमें वो क्रिएट किया जिसकी वजह से क्या हुआ ऑटोमेटिकली जो हमारे सेंट्रल का ब्लड सर्कुलेशन है वो क्या हुआ रिड्यूस हुआ. इसके अलावा अब रिसेंट कुछ स्टडीज ने ये भी चीज बताई है कि क्रोमेटिन जो हमारी बॉडी के अंदर ल्यूकोसाइड एडेशन मॉलिक्यूल्स होते हैं, ठीक है ना, इंट्रासेलुलर एडेशन मॉलिक्यूल्स होते हैं, उनके एक्सप्रेशन को भी क्या करता है कम करता है जिसकी वजह से क्या है इन्फ्लेमेटरी पाथवेज वो ट्रिगर होते हैं जैसे ल्यूकोसाइड एडेशन, ओके, ल्यूकोसाइड इन्फिल्ट्रेशन, ऐसे फिनोमेना बहुत ज्यादा क्वांटिटी में इंक्रीज होते हैं और ये बाद में क्या करते हैं ल्यूको इन्फ्लेमेटरी कास्केड्स को फर्दर एक्टिवेट करते हैं. और भाई रिसेंट स्टडी अभी 2026 में एक रिसेंट स्टडी आई है उसने ये भी बताया है कि जो क्रोमेटिन है इट प्राइमरीली इंड्यूस्ड इस्कीमिया इन ह्यूमन बाय टारगेटिंग द TNF अल्फा रिसेप्टर्स. जब भी TNF अल्फा रिसेप्टर्स को टारगेट करता है उसकी वजह से क्या होता है कि न्यूरो इन्फ्लेमेटरी कास्केड्स बढ़ जाते हैं एंड न्यूरो इन्फ्लेमेशन इज वन ऑफ द मेजर पैथोलॉजिकल इवेंट दैट अकर इन द cerebral ischemia. Next slide please. So why this is we are discussing why chromatin. सबसे पहले इसके जो कॉमन फीचर की बात करते हैं ड्रग इंड्यूस्ड मॉडल है, हमने पहले भी डिस्कस किया. अभी तक कोई भी ऐसा ड्रग इंड्यूस्ड मॉडल या केमिकल इंड्यूस्ड मॉडल अभी तक किसी भी स्टडी में या कोई भी प्री क्लिनिकल एविडेंस इसके लिए यूज नहीं है, ना ही इसके बेसिस पे अभी तक जो है ड्रग टेस्ट की गई है प्री क्लिनिकल फेज के अंदर. तो बेसिकली इट इज एन हाईली रिलायबल मॉडल कि हम सेकेंड टॉक्सिसिटी जो है प्रोड्यूस कर रहे हैं. सेकंड पैथोफिजियोलॉजिकल मैकेनिज्म, it produces the same pathological mechanism that is being produced to the human model of cerebral ischemia. Second model advantage है अगर हम बात करते हैं rodents के अंदर भी, तो rodents के अंदर जो हम cat के अंदर या mice के अंदर जो हम अपना क्या कहते हैं ischemia induce कराने के लिए जो मॉडल यूज करते हैं इट्स प्राइमरीली इन्वॉल्विंग सर्जिकल प्रोसीजर्स. मिडल सेरेब्रल आर्टरी कल्चर मॉडल चाहे वो लियाजोना मेथड का है या कोइजोमी मेथड का है हम उससे प्रोड्यूस करते हैं बट वो एक सर्जिकल प्रोसीजर है. तो हर कोई इंडिविजुअल इज नॉट ट्रेन्ड फॉर द सर्जिकल प्रोसीजर्स. तो इट इज वन ऑफ द नॉन सर्जिकल प्रोसीजर्स एंड हाईली रिप्रोड्यूसिबल एज कंपेयर्ड टू एनी अदर मॉडल क्योंकि इसके अंदर मोर्टेलिटी बहुत कम है. वन इज द ट्रांसलेशनल एलिमेंट्स क्योंकि अगर हम बात कर रहे हैं किसी क्लिनिकल स्टडीज के लिए तो ट्रांसलेशनल एलिमेंट्स में बात आती है कि कितना वो उसको हम क्या कहते हैं एक्यूरेटली रिफ्लेक्ट करेगा हमारे मेडिकल डिसीज की पैथोफिजियोलॉजी को. स्क्रीनिंग यूटिलिटी कॉस्ट इफेक्टिव है, हमने पहले डिस्कस करा. एक साथ क्या कहते हैं बहुत सारे हम ड्रग्स को या वैस्कुलोप्रोटेक्टिव
न्यूरोप्रोटेक्टिव एजेंट्स को साइमल्टेनियसली हम क्या कहते हैं इवैल्यूएट कर सकते हैं, सेम पैथोफिजियोलॉजिकल रिलेवेंस है और ब्रीथ बेसिकली रिसर्च की तरह जो रिसर्च complex mammalian models thereby improving translational analytics like this. So what you do, we will just this is the one of the most monitoring mediated ischemic damage in our adult geography. हमने क्या किया, हमने बेसिकली 4-5 डिफरेंट कंसीडरेशंस की monitoring की 2.0.25 mg per liter से लेकर हमने 2 mg per liter तक हमने क्या किया monitoring की एक्सपोजर किया अपनी जेब्राफिश को फॉर दैट जेब्राफिश हमने उनको 30 मिनट्स का एक्सपोजर दिया वाटर इमर्शन मेथड से। फिर हमने क्या किया 24 आवर्स तक उनको कंटीन्यूअसली ऑब्जर्व करा फॉर एनी काइंड ऑफ बिहेवियरल चेंजेस उनके मोर्टेलिटी को देखी और आफ्टर द 24 आवर्स हमने क्या किया उनकी ब्रेन डैमेज देखी बाय टीटीसी स्टेनिंग कि उनके ब्रेन के अंदर कितना डैमेज हुआ। नेक्स्ट स्लाइड प्लीज। सिमिलरली हमने क्या करा अपने मॉडल को कि अभी तक जो है हाइपोक्सी इज वन ऑफ द वैलिड मॉडल फॉर इस्कीमिया इन एडल्ट जेब्राफिश। तो हमने हाइपोक्सी को अपना एज अ रेफरेंस मॉडल मान के चला कि हम कंपेयर करते हैं कि रिजल्ट कैसे है। हमने हाइपोक्सी कैसे यूज किया कि हमने सिंपली एक एयर टाइट कंटेनर के अंदर अपने फिश की वाटर टैंक के अंदर हमने जो है वहां पे डिसॉल्व्ड ऑक्सीजन का लेवल 7 to 8 mg per liter जो होता है उसको रिड्यूस करके 0.8 mg per liter किया, उसको 30 मिनट तक हमने हाइपोक्सी कंडीशन दी। देन we can say that हमने क्या किया उनको फिर उनकी हाइपोक्सी प्रोग्रेशन को देखते हुए क्या किया ट्रांजिट इस्कीमिया को उसके और फिर हमने उसको नॉर्मल जो हमारे क्या कहते हैं टैंक से उसके अंदर ट्रांसफर किया जिनका डिसॉल्व्ड ऑक्सीजन लेवल 7 to 8 mg per liter था। So इस तरीके से उनके अंदर भी इस्कीमिया इंड्यूस हुआ बट this kind of ischemia is primarily global type of ischemia, not focal type of ischemia. But we are not any specific model for focal type of ischemia. तो हमने अपने हाइपोक्सिया मॉडल तो हमने अपने क्या है हाइपोक्सिया के मॉडल को क्या है एज अ रेफरेंस स्टैंडर्ड अपने स्टडी के अंदर यूज किया। नेक्स्ट स्लाइड प्लीज। देन दिस इज द बेसिकली रिजल्ट्स की 24 घंटे के एक्सपोजर तक हमारी जो हमने हाइपोक्सिया इंड्यूस्ड फिशेस थी या डिफरेंट कंसंट्रेशन ऑफ पोनेटिक इंड्यूस्ड हमारी जो जेब्राफिश सबकी हमने प्रोबेबिलिटी ऑफ सर्वाइवल देखा। तो हमने ये चीज देखी कि कंपैरेटिवली हाइपोक्सिया के अंदर एट अ अर्ली स्टेज मोर्टेलिटी आने स्टार्ट हुई एंड बाय द 24 आवर्स अराउंड उनमें 80% तक मोर्टेलिटी है बट हमारे जो कहते हैं पोनेटिक एक्सपोजर उसमें मोर्टेलिटी कंपैरेटिवली बहुत कम है। अगर बात करें 0.25 mg per liter 1 mg per liter 2 mg per liter में कंपैरेटिवली बहुत कम हमारी क्या है प्रोबेबिलिटी ऑफ सर्वाइवल प्रोबेबिलिटी ऑफ मोर्टेलिटी है मतलब कि 50% अभी तक हमारे क्या है आफ्टर 24 आवर्स हमारी जो जेब्राफिश सर्वाइव है। उसके बाद हमने क्या करा जो हमारी इसी पार्ट में है हमने उनके बिहेवियर देखे कि जो एक स्कीमिक कंडीशंस के अंदर सबसे पहले उनके अंदर क्या है बिहेवियर चेंजेस ऑब्जर्व होते हैं। तो हमने देखा अब ए में देखें तो अगर बात करें दैट दे स्टार्ट स्विमिंग एट द टॉप उनके बिहेवियरल इंपेयरमेंट है कि नॉर्मली फिश क्या करती है अपने वाटर एनवायरनमेंट के पास ही मूव करती है बट जब उसके अंदर कोई भी स्ट्रेस लाइक कंडीशंस आते हैं तो क्या करती है पानी से बाहर निकलने की कोशिश करती है। तो दिस इज द कंडीशंस की स्विमिंग एट द टॉप एंड आफ्टर few minutes we will show the fish just stand like a simple vertical position में आके stand होगी but it start losing its position जो उसके buoyancy की बात करते हैं fish की start losing its buoyancy थोड़े टाइम बाद उसको क्या कहते हैं जो उसकी ocular beats होती है कंपैरेटिवली जो उसकी हार्ट बीटिंग होती है वो काफी ही स्लो होनी स्टार्ट होती है और आफ्टर सम टाइम इट विल शो दैट। तो सेम हमने क्या देखा कि हाइपोक्सिया मॉडल के अंदर भी और पोनेटिक इंड्यूस्ड मॉडल के अंदर भी हमें सेम 4 डिफरेंट टाइप्स ऑफ बिहेवियर इनपुट दिखे बट जो हमारी डेथ थी कंपैरेटिवली हाइपोक्सिया मॉडल के अंदर जल्दी आई बट जो हमारी डेथ है जो बात की हमारी पोनेटिक इंड्यूस्ड मॉडल में कंपैरेटिवली बहुत लेट आई और जो सिमिलरली बिहेवियर इंप्रूवमेंट्स हमें दिखी वो बिल्कुल एक तरह से बात नहीं थी सिमिलर टाइम पैटर्न पे हमें उनके बिहेवियर चेंजेस दिखे। देन वी विल गो फॉर द brain damage हमने उनकी टीटीसी स्टेनिंग करी वन ऑफ द स्टैंडर्ड प्रोटोकॉल टू डिटरमाइन दिस इस्कीमिया। हमने उन्हें देखा कि उनका टोटल कितना ब्रेन डैमेज हुआ है तो हमने देखा कि जो हमारी कंट्रोल फिश है उनके अंदर टीटीसी स्टेनिंग की कंसंट्रेशन अगर बात करें 100% थी तो हमारी हाइपोक्सिया के अंदर इट रिड्यूस टू अबाउट 48% बट जो हमारी ड्रग है उसमें सिमिलर काइंड ऑफ जो है अपने क्या कहते हैं हाइपोक्सिक कंडीशन सॉरी इस्कीमिक कंडीशंस के अंदर टीटीसी स्टेनिंग के फैक्टर से जो है अपना इस्कीमिक रीजन को शो किया। तो इस बेसिस पे हमने देखा जो हमारी पोनेटिक 1 mg per ml है इट विल शो वन ऑफ द बेस्ट ऑप्टिमाइज्ड डोज जिसके अंदर हम क्या कह सकते हैं कि हमारी जो ड्रग है या पोनेटिक की बात करें हमारी डोजिंग मॉडल है उसने क्या किया अपनी क्या कहते हैं इस्कीमिक लाइक पैथोफिजियोलॉजी एट दिस कंसंट्रेशन उसने शो कर दिस इज बेसिकली कंपैरिजन दैट जो हमारी पोनेटिक इंड्यूस्ड मॉडल है उसने ऑक्सीडेटिव स्ट्रेस को भी बढ़ाया माइटोकॉन्ड्रियल डैमेज भी करा, थ्रोम्बोसिस की जो हम बात करें जो वन ऑफ द मेजर पैथोलॉजिकल हॉलमार्क ऑफ दी स्कीमिया वो भी इसने प्रोड्यूस किया, देन इट विल आल्सो इंड्यूसेस न्यूरो इन्फ्लेमेशन एंड द वन ऑफ द लास्ट इंपॉर्टेंट फैक्टर दैट इट इंड्यूसेस द न्यूरोनल ग्रोथ फैक्टर्स एज वी डिस्कस्ड अर्लियर कि इसके अंदर वेस्कुलर एंडोथेलियल ग्रोथ फैक्टर्स के एक्सप्रेशन है वो क्या होते हैं, कम होते हैं। सिमिलरली जैसे हम बात करते थे कि एमसीओ पेशेंट के लिए सेम लाइक ऑफ फिजियोलॉजी हम बात करते हैं प्रोड्यूस होती है। नेक्स्ट स्लाइड प्लीज। So what are the basic conclusions? He that this study successfully established this as a model, one of the reliable model to produce ischemia. It significantly induces brain injury to reduce behavioral impairments as shown in these figures. Then we based upon that we found that Ponatinib at a concentration of 1 mg per liter produced an optimal ischemic like neurological defects. And one of the major that it has the comparative analysis हमने अपने hypoxia model से करी तो हमें ये भी चीज पता चली कि हाइपोक्सिया मॉडल में कंपैरेटिवली हमारे इस मॉडल के अंदर मोर्टेलिटी कंपैरेटिवली बहुत कम है, सो इट ऑफर्स एज अ रोबस्ट एंड रिप्रोड्यूसिबल प्लेटफॉर्म। नेक्स्ट स्लाइड प्लीज। अगर हम इसके ट्रांसलेशनल आउटकम के बारे में बात करते हैं, सबसे पहली बात की इट एक्ट एज एन अल्टरनेटिव टू हाइपोक्सिया बेस्ड पैराडिज्म, रैपिड स्क्रीनिंग हम एक साथ बहुत सारे ड्रग्स को स्टडी कर सकते हैं, सिंपलीफाइड एक्सपेरिमेंट प्रोसीजर्स किसी भी तरह के हमें हाइपोक्सिया के लिए हमें ट्रीटमेंट की जरूरत होती है, हमें ऑक्सीजन मॉनिटर की जरूरत होती है, बहुत सारे डिफरेंट हमें इंस्ट्रूमेंट्स की जरूरत है। हाइपोक्सिया पोनाटिनिक इंड्यूस मॉडल के अंदर हमें ऐसे किसी भी इंस्ट्रूमेंट की स्पेशलाइज्ड ऑपरेटर्स की जरूरत नहीं है। एनहांस एक्सपेरिमेंटल रिप्रोड्यूसिबिलिटी एंड स्केलेबिलिटी, क्योंकि हम पोनाटिनिक को लेके हम अपने क्या कहते हैं बेसिकली 6 वेल्कुलेट के अंदर ही हम क्या कहते हैं अपनी स्टडी को हम क्या कहते हैं टेक ओवर कर सकते हैं। देन प्रोवाइड्स अ प्रैक्टिकल प्लेटफॉर्म, क्योंकि हाइपोक्सिया करने से बेटर है कि हम उनमें केमिकल इंड्यूस इस्कीमिया का है। ओके, तो अपने आप में क्या कहते हैं कि ट्रांसलेशनल पर्सपेक्टिव है। इट आल्सो सपोर्ट्स अर्ली स्टेज डिस्कवरी एंड डेवलपमेंट को। अब जो मेजरली जो हम बात करते हैं कि रोडेंट मॉडल्स के अल्टरनेटिव को ढूंढा जा रहा है, तो अपने आप में जो यूज ऑफ वेल जेबरफिश एज अ मॉडल एक रिलायबल मेथड है। नेक्स्ट स्लाइड प्लीज। सो दीस आर द सर्टेन रेफरेंसेस दैट आई यूज्ड ड्यूरिंग दिस। थैंक यू वेरी मच।
Okay, Karan. Any questions from the participants? Sir,
Hello, Mr. Karan Wadhwa.
I started the presentation. So, when you establish this model, okay, how many times you tried, you repeated the your experiments to validate? How did you validate this model?
Basically, I have performed this model around 10 to 12 times. मैंने सर हर बार ही जब मेरे पास पोनाटिनिब आया, सबसे पहले तो सर मैंने उसको ऑक्सीजन देखी थी। एक्चुअली अगर मैं जो रिपोर्टेड लिटरेचर है कि उसके बेसिस पे जो मैं कंसंट्रेशन जो मैंने 0.12 mg पर लीटर तक अपनी डोज चूज कर ली, ऑरली भी आईपी देनी है या किस तरीके से देनी है, बाटा इमल्शन देनी है, तो सबसे पहले सर हमने ये प्रोसीजर चेक करा। तो इसमें भी हमने यूज बट जो आप क्वेश्चन पूछ रहे हो कि हमने कितनी बार मॉडल को अपने लैब में कितनी बार हमने किया, कंपैरेटिवली सर हमने उसको अपनी लैब में 10 से 12 बार करा। उसके बाद सर हमने अपने कुछ ड्रग्स भी क्या कहते हैं जो हमारी न्यूरोप्रोटेक्टिव ड्रग्स थी उनकी एक्टिविटी भी एज इसको अपने क्या कहते हैं अपना नॉर्मल की यूज भी की। 10 से 15 बार से हमने इसको अपनी लैब में यूज किया है फॉर की वैलिडेट करने के लिए कि हम जो बारी बारी अपने काम को रिप्रोड्यूस कर रहे हैं या टर्मिनोलॉजी की वो रिप्रोड्यूसिबल रिजल्ट दे रहे हैं नहीं दे रहे। सेम सर इसका एक और भी है कि हमने अपनी लैब में तो काम किया ही किया, इसके अलावा सर एक दूसरी लैब में भी इसके ऊपर काम अभी करना स्टार्ट कर दिया, उन्होंने भी यूज किया है एंड दे आर गेटिंग सिमिलर रिजल्ट दैट
Okay, very good.
वही पावर बैंक चल रहे हो।
थैंक यू सर।
माइक उठा लो।
Okay, Karan. Thank you.
चार्जर
Now we request Ms. Vijeta Sharma, Senior Research Scholar, Department of Cell Culture, Noida. Ms. Vijeta Sharma.
डाटा केबल नहीं है दूसरी, एडल में दिक्कत हो रही मे भी, पता नहीं शायद ये और इस वाले फ़ोन करके देखे जाने को डाटा केबल में दिक्कत हो रही, ठीक है तो ये है तो लोकल डाटा केबल चेक के साथ ये हाँ
Now, Dr. Sharma, Senior Research Scientist, Department of Cell Biology, DAFA Research, India, will be our next participant in this today's oral presentation competition in this 2-day international conference sponsored by UPCST.
Good afternoon, everyone. I would like to thank SD College of Pharmacy and Vocational Study for inviting me here to present my work. Today, I am going to present my work on evaluation of hepatoprotective activity of herbal formulations against CCL4-induced experimental hydrotoxicity using male starlight. What was the rationale of work? As we know, liver disease is still a worldwide health problem, but unfortunately, conventional or synthetic use in the treatment of liver are inadequate or sometimes can cause serious side effects. So, as per the study, no work has been done yet on the combination of pipernigram, gingiva officinae, and moringa oleifera, which three are herbal drugs. All of the above possessing antioxidant activity, so it can be a good lead to develop a hepatoprotective drug. Because as we know, hepatotoxicity का main cause जो है वो oxidative stress होता है. So reducing oxidative stress can be helpful. So the aim is to evaluate and compare the hepatoprotective activity of formulations containing pipernigram, gingiva officinae, and moringa oleifera in different ratios, that is 1:2:4 and 1:3:9 ratio respectively. Introduction of liver. Liver is the largest organ located in the quadrant of abdominal cavity below diaphragm. Role is maintaining, performing, and regulating homeostasis of the body, involved in various biochemical pathways of growth, fight against disease, nutrient supply, and energy provision. Vital functions of the organs are listed here: excrete waste and bile. It is a factory of protein and cholesterol. Excrete bile for fat digestion, regulate blood clotting, convert excessive glucose to starch for storage. It also helps in clearing blood drug chemicals and alcohol, immunity against infection. Next slide, please. What is hepatotoxicity? Hepatotoxicity refers to as liver dysfunction or liver damage that is associated with an overload of drug or xenobite. Xenobite is anything which is toxic to our biological system. And what are the causes of liver toxicity is mentioned here. If we talk about the chemicals, various chemicals are available which can be used to develop a hepatotoxic models like CCL4-induced hepatotoxicity, phosphorus, aflatoxins, and chlorinated hydrocarbons. Other than this is alcohol. About 50% of the liver toxicity is caused by consuming alcohol. Other than this are some viral infections like hepatitis A, B, and C, autoimmune disorder, and drug-induced liver injuries. Next slide, please. pathway by CCL4 induced hematotoxicity in one liver. While we inject CCL4 induced, it gets converted into the carbon trichloride, which ultimately lead to the formation of ROS, that is reactive oxygen species, which will lead to increase in the oxidative stress. As oxidative stress is increased, there is rise in the various hepatotoxic parameters like SGOT, SGPT, GGT, ALT, bilirubin T and bilirubin T. Other than this, liver damage, it also goes to liver damage, that is fibrosis and inflammation. So our main goal is to decrease these free radicals or suppress these radicals by using antioxidant products, so that we can reduce all these, we can decrease all these parameters to our therapeutic level. As you can see here, antioxidant and hepatoprotect reduce the oxidative stress and ultimately leading to decrease in the SGOT, SGPT, DGT, ALT, bilirubin T and D. Also, it also decreases the liver damage. So this is the pictorial diagram which is representing the methodology, like how we conducted the experiment. First of all, we prepare herbal formulation, and I have already mentioned the name of herbs I have used. Preparation of the herbal formulation. Next part comes is animal dosing. Other than again disease induction, that I have already mentioned that we have induced the disease by using CCL4 injection. Then collection after completing of all the dose regimen, collection of the blood sample, necropsy. After that, biochemical analysis, histopathology analysis, and finally to the data analysis.
ठीक हो
So herbal formulation. Herbal formulation is the dosage form of one or more herbs possess herbs in particular quantity to provide specific nutrition. Like formulation used in this study is mentioned here. I have used two formulations, formulation 1 and 2. Herbs are same, the only thing which vary is the ratio of these herbs. In formulation number 1, I have used pipermigran, jungivar officinale, moringa oleifera in 1:2 ratio, and in formulation 2, I have used it in 1:3:9 ratio. Next slide, please. Okay, grouping of animals. So basically, this is the design which I have used for conducting the study. Many of the students must be planning their M.Pharm project and they must be confusing how to design a model for in vivo studies. So this will be helpful for them, like how they can design and conduct their experiment. Here I have used 5 groups. In G1 is the normal control group. It means that no treatment was given to this group. In this group, doses and route of administration was 10 ml per kg, that was used orally. Regimental duration was daily for 28 days, and the number of animals was 6 animals per group. Second group was disease control group. In this, I have injected CCL4 injection in olive oil, and the regimen was 10 ml per kg orally, weekly twice, daily for 28 days, 6 animal was there in this group as well. Other than this, G3 was the positive control group. Here, disease induction was same, olive oil, CCL4 injection in olive oil, but the drug was Silmarin, that already marketed drug available in the market for the hepatic activity. G4 and G5, the group of interest for us, that is formulation A and formulation B. As I have already mentioned, formulation A was, sorry for this typographical error, formulation A was 1:2:4, and formulation 2 or B was 1:3:9, and regimen was 28 days, daily decimal per group. Next slide, please. So this was the hepatotoxicity induction and treatment regime. In green grids mention the dosage induction of our drug, and red block mention the drug injection along with the CCL4 injection. Like at the alternate days, we have injected CCL4 to the animals, and in the green grid I have mentioned device formulation we have injected. Here you can see treatment for green, disease induction plus treatment for red blocks. Okay, result. Here all the animals were observed for clinical signs and symptoms throughout the study. No mortality was observed throughout the study. The hepatoprotective effect of formulations was evaluated by measuring various parameters like SGOT, SGPT, GGT, ALP, bilirubin D, D, and C reactive protein. Hematological estimation was done by using WBC and lymphocyte count. Body weight was measured, relative liver weight was measured, and histopathology was performed. Next slide. So here are the results of my experiment, where you can see the difference. G1 was the normal control group. As you can see that the level of SGPT is very low in G1 group, but in the G2, that was my disease control group, it get increased significantly. And along with this, you can see G3, G4, and G5. There is a significant decrease in the levels of these parameters. Basically, SGPT and SGOT are the parameters or the markers of hepatotoxicity. It indicates the damage of cells. So, decreasing the level of these in blood is our ultimate goal. And using this formulation, we have achieved that successfully. So this is the effect of oral formulation on level of GGT and ALPs. Again, these were also the marker of hepatotoxicity, like if our liver is damaged or the cell death is occurring, the level of GGT and ALP will get increased. And you can see here, in the group 2, the level of these GGT and ALP is very high. But as we given the therapy or treatment, it gets decreased significantly. Next slide. Similarly, bilirubin T and bilirubin D also showed very good effect while using our formulations. Next slide. So this was the hematological parameters which we have checked. Here WBC count and lymphocyte count was checked. As we all know that WBC and lymphocyte counts are the markers for inflammation in our body. And you can clearly see that in G4 and G3 the level of these both markers are low as compared to our disease control group. And you will be surprised to know that the result was better than the Silmarin as well. So this is the CRPs. Again, CRP is also a blood marker for hepatotoxicity, that is C-reactive protein. Here also the results were very promising, and you can see the different significant decrease in the level of these markers in blood significantly. Next slide. Next slide, please. So this was the relative liver weight. As we all know that during the hepatotoxicity or any disease, we is, as you can see, this is the normal architecture of a liver, of a scar. Here you can see there is a centrilobular necrosis and inflammation in his control group. Here Silmaric again, written again the liver structure, architecture of liver structure retained to its normal condition. And here you can see that difference as compared to the CCL control group. Formulation A is showing very good effect as compared to the disease control group. So, formulation A showed a marked improvement in the hepatocellular architecture and was found superior in hepatoprotection as compared to formulation B or Silmaric to the CCL4 treated rats. So, the aim of study was to evaluate and compare the hepatoprotective activity of formulation A and B containing pipernicum, gingiber officinale and Moringa oleifera in 1:2:4 and 1:3:9 ratio respectively. In the present study, CCL4 induced hepatotoxicity model was used to investigate the hepatoprotective potential of this formulation, and administration of CCL4 results in marked elevation of enzymes like SGOT, SGPT as a result of cell lysis and necrosis, and marked elevation of serum ALT level, which represent the alteration in the integrity of hepatobiliary system. Treatment with these formulations reveals the elevated level of these enzymes, indicating hepatoprotective property of these formulations. Next slide please. So, in the present study, the administration of formulation A and B to the hepatotoxicity reduced the development of inflammation in CCL4 induced by serum CRP level. The concentration of black pepper as a 1 part in formulation A was relatively more than in formulation B, suggesting that on the basis of bioavailability enhancer, anti-inflammatory and antioxidant property, black pepper may be considered as a key component of any formulation for management of the oxidative stress and inflammation related chemicals. So, taken together, you can say that overall observation estimate that hepatoprotective potential of formulation A, that is in ratio of 1:2:4, was better than the formulation B, showing no additional benefit beyond 1:2:4. So these are some references which I have used during my work. Thank you.
Thank you, ma'am. Volunteer, any volunteer, please come up. Shami, please come here. Hello. I request any queries, any questions from the audience. Hello, please.
Ms. Vijayta Sharma, your attendance online. I have some questions. What rationale was used in selecting the combination?
So we have done a thoroughly literature search. If we talk about the moringa was in very high concentration in all these formulations. As we go through the literature, we have seen a good activity as a hepatoprotective activity of moringa oleifera. But on the same time, we could see in the literature that there was a bioavailability thing which was coming in between the activity of these things. So, as per the literature search, black pepper was showing very good effect when we talk about the bioavailability. So we have chosen this, and same thing with the gingiber officinale.
When are you talking about bioavailability? Did you focus on particular phytochemical inside these two drugs?
No, sir. we have
Then
used.
how can you say the pipeline diagram was used to enhance the bioavailability?
Sir, we have done some studies for the pipeline as well. The result is not mentioned here, but the pipeline, the key compound of piperidine is responsible for enhancing the bioavailability.
Okay. One more question. You used the two combinations, formulation A and formulation B. What criteria you adopted in selecting the drugs in this particular combination?
Sir, so it is depend on the safety of each house, as the piperidine is highly toxic, and on the same time the moringa was safe as well. So if we have chosen the concentration based on the toxicity, like we have done the MPPS, safe dose for this. Piperidine was highly toxic and in the hepatic cells, and so we have choose very low concentration of these just to enhance the viability, not for biological action.
Okay. Another question. Why did you opt CCL for induced hepatotoxicity model? Why not other?
So actually we have tested the ethanol model as well, but the drawback with the ethanol model is result is not consistent. Like if we talk about the disease model, if we are choosing 6 animals each group, the level of SGPT and SGPT in each animal was very different. So the consistent result we could see only in the, we have tried both, but the consistent and like we can say we were confident only with the CCL is being used for.
Okay. It's fine from my side.
Jayla,
Yes, sir.
according to you, formulation 1 is better than B.
Yes, sir.
Can we correlate formulation 1 with fat metabolism?
For
Fat metabolism.
fat metabolism.
Ji.
No, sir, we have not done this study.
Oh.
Okay.
Madam, have you done the docking studies?
No, sir. It
It
was
was for
only
the
the
docking studies also.
Yeah, we can do that, but we have done only the in vivo studies.
And what was the solvent for the taking these extractions of the drugs?
0.5% CMC.
Okay.
Yes, sir.
Thank you. Just a minute.
हेलो
Yes, ma'am.
मैम पहले तो आपकी बहुत प्रेजेंटेशन अच्छी लगी, थैंक यू। मैम आपने इन विट्रो सेल लाइन स्टडी की है इसकी, या आपने अभी जो आपने काम किया है वो सिर्फ इसके बेस पे किया है इन विवो?
Like, we have done the study on HG2 cell line.
HG2 आपने किया है, तो मैम आपने ये डाटा यहाँ पे शो किया है?
No.
आपने शो नहीं किया, बट यू हैव डन
Yes.
ऑन द इन विवो सेल लाइन स्टडीज।
MTT studies we have done just to determine the safe dose. Like we were giving these formulation to animals to check before we have done the MTT.
And ma'am, why you choose only HG2 cell line?
See, we were doing our study on for hepatotoxicity model. So HG2 cell line is the liver cell line, so it makes sense.
एंड मैम आपने जो मार्केटेड प्रिपरेशन ली है इसमें कंपैरिजन के लिए वो क्या है मैम आपका? सिलीमेरी। तो ये मार्केटेड है?
Yes, it is already selling in the market.
ओके, तो मैम ये जो इंडिविजुअल था जैसे आपने पाइपर नाइट्रम लिया है और अदर टू लिए हैं, तो इंडिविजुअल भी हो रहा होगा इनका?
No, we have not done for individual. Actually, we were thinking of developing a formulation. So we just did for the formulation. Actually, we have done the study on about this concentration as well, but the result it was not showing good. The variation in the result was very high. So yeah.
नहीं मैम, इनकी इंडिविजुअल भी स्टडी हो रही होगी जैसा आपने लिटरेचर किया होगा।
Yes.
तो उसके बाद आपने इनको कॉम्बिनेशंस में लिया है, डिफरेंट वेरिएशंस
Yes, it is already published in various literature. They like all these three herbals having very good hepatoprotective activity, but no one has done in the combination.
एंड मैम have you done the LFT for this liver function test?
Liver function test
SGLT
is all LFT only.
and lipid profile?
Sorry?
Lipid profile?
No.
Lipid profile?
Lipid profile.
No, you haven't done? No. Okay. Thank you so much, ma'am. Thank you.
Thank you so much. Now request Ms. Aditi Yadav from TMU Muradabad for the next presentation. Ms. Aditi Yadav. Is she available? Ms. Aditi Yadav, please. She is from TMU Muradabad and one of our participants of oral presentation. Aditi.
Good afternoon, everyone. My name is Aditi Yadav. I am a PhD scholar and also a junior research assistant under the project of UPCSD. And I am from T. Thankar Mahavir University. It's a graphical abstract and which shows the significance of plumbago species and also known as chitra, which focuses on historical use of traditional medicines to the potential for global therapeutic applications. Introduction: in recent years, there is an increase in the many disease, which leads to the decline in public trust with the modern disease, the modern medicines, and renaissance in the use and study of plant-based and traditional therapeutic methods. So my plant is chitra and also known as plumbago zelenica, which is fall under the category of Plumbaginaceae, and it is also known as Dr. Bush, where the plumbago is comes from the Latin word plumber, which means lead, and zelenica means cell, and it is having many used in the treatment of many diseases like pains, removal of intestinal parasites, leprosy, ulcers, rheumatitis, inflammation, and here I am discussing about the many other different species of plumbago: Plumbago zelenica, Plumbago indica, and Plumbago auriculata. The flower of Plumbago zelenica is pure white, indica vibrant red, and auriculata serene blue. Height is different: 9 feet tall, 5 feet tall, and 5.5 feet tall. This is the core phytoconstituent, which is plumbagin, with different higher concentrations. Highest concentration in Plumbago zelenica 0.1 to 0.6%, in indica 0.05 to 0.2, and in auriculata is very low, like in trace amount. Geography: Africa, Middle East, Second India, South Africa, and this is the Plumbago zelenica indica and auriculata. This is the use of plumbago across traditional systems like in Ayurveda, Siddha, Unani, and folk. Chitra is known as in Ayurveda, which is used in digestive stimulants and carbonated treatment of indigestions, and common preparations are dry or decoctions. Next is the chitra, gastrointestinal disorders or in skin diseases, components of formulation for certain tumors and cancers, and common preparations are decoction, paste, or processed roots. In Unani, it is also known as citrage and chitra, stimulants, carbonative, alternative, or digestive complex, and common preparations are also decoctions and extracts. So in folk, chitra is a lead good. It is used for antipyretic, antimicrobial, or antiplasmodial, anti-inflammatory. Common preparations are root fructose, phosphate, 4 expression, and in anti-hyperlipidemic activity, it decreases the cholesterol and decrease triglyceride. In antibacterial activities, decrease effective against MRSA, increase MRC, and increase 99% to kill in 8 hours. In anti-malaria activity, decrease SDH enzymes, decrease parasite growth. These are the modern drug deliveries of my plant of Plumbagis indica or Chitra. First, for diseases, antibacterial nanoformulations, and this is the outcome. And for acne, gel is formulated. For anti-diabetic tablet, for anti-diabetic and antioxidant soaps, and for anti-rheumatic microemulsions are formulated. And these are the modern drug delivery system other than Vati and other powders. Toxic effects of this plant is burning sensation in the tongue, throat or stomach, nausea, vomiting, diarrhea, weak pulse, dermatological manifestation like wrinkled or cold skins. Treatment is lavage of stomach with water or water. Depending on the symptom, administering stimulants and other alternative drugs, use of oxygen, artificial respiration, antidote, preparation of pitas, shamakas, shita, conventional propagation. These are the conventional propagation, seed propagation, vegetative propagation. These are done by the seeds and stem. This is a tissue culture strategy, also known as micropropagation. Micropropagation, campus suspension culture, here culture, somatic invertebrates. This is the medium used in outcomes. Commercial products. Many commercial products are available in the market, like Chitrakati Bati, Harikati, Chitrakati Gutika, whose brands are Lava, Ernath and Plant Ayurveda. Single ingredient raw botanical products like dried root powders, which is available on Amazon and other websites. Nutraceutical herbal supplements, commercial capsule and tablets are also available for the treatment of digestion and appetite. Next, Dabur Chitrak Haritaki, Vaidnath, Plant Ayurveda. These preparations are. There are some patents of this plant also. Preparation extraction, like Chinese patent is using a method for preparing and purifying combagin. Next, therapeutic composition, small molecule derivatives, claims of combagin derivatives. Lastly, veterinary agriculture composition. These are relevant for livestock and poultry disease. This is the conclusion. It demonstrates a significant relationship between the different species of the plant, which is not only useful for the botanical insight, but also for the pharmacological actions. And the global significance of Chitrak is shown by this synthesis of traditional knowledge and contemporary studies, which is helpful for the future perspectives. Thank you.
Hello. Aditi,
Yes.
your presentation was good. Okay. You gave a thorough literature about the flamenco species. As you mentioned that one of its main bioactive compound is plumbagin. Did you gather information about its bioavailability?
So not now, but I'm working. It is just review, so I'm working on it. I'm doing it.
Because as you mentioned, a number of marketed formulations containing this extracts or maybe plumbago might be one of the constituent in those formulations, say gel, nano-emulsions, neosomes. Okay. So, can you tell me if any of its formulations of this molecule is approved by any regulatory body?
Sir, I have not studied that far because my work is just on the identification. My PhD work is just on the identification. I am not working on that.
But it will be worth if you gather this information, include this information also. Okay, bioavailability, okay, and it's more over this plumbago. Any toxicity related to this plumbago? Any information related to toxicity?
Sir, it is quite toxic if we are not doing Shodhana. Shodhana means purifying the stomach.
Shodhana means purifications.
So after purification we can use it without that is it is not having that toxicity, but we can do.
Shodhana is an Ayurvedic method for purifying the drug, for detoxifying the drug. Okay, but any toxicity study reported on this compound? Can you? Okay. If you include the toxicity profile also, it will be a good addition. Thank you. Any other?
Hello Aditi. Hello ma'am. You present so nice. Your confidence. It's my small suggestion for you. Whenever you present your presentation anywhere, make sure you have write the page number on each and every slide. ताकि हमारे लिए ये easy हो कि पूछना कि किस slide पे क्या है. And one of your slide you use the term MIC. MIC. What is the meaning of MIC? Actually, I have not. No, you use the MIC term. I want to know what is the full form MIC, only full form. You use in antibacterial activity. And you know this, MIC is 2.5 or whatever it is microgram per ml. So what is MIC? No. Okay, no problem. How you differentiate Ayurveda, Unani and Siddha? A small difference only. In Ayurveda, we work on the 5 pitas. What is going under our body? Like pitta जो होता है, earth, water, air and fire. And in Unani we work on the symptoms. And Siddha, I think it is also quite related to symptoms. To use and plumbago is used as antibacterial or other. You have mentioned what is it? Other than antibacterial, anti-malarial और भी कुछ था. ऐसी कौन सी मोइटी थी जो एंटीबैक्टीरियल के लिए रिस्पॉन्सिबल थी? Plumbagin. So it is flavonoid, triterpenoid. What is this? Ma'am, it is naphthoquinone. Sorry, naphthoquinone. Naphthoquinone. And in anti-malarial, ma'am, actually, plumbagin is working in all, and it is a naphthoquinone. Its category is naphthoquinone. मार्केटेड प्रिपरेशन का नाम जिसमें प्लम्बैगो है, मैम हाँ, चित्रकटी और चित्रक हरी टकी. तो चित्रकटी किसके लिए यूज हो रही है? मैम इट इज यूज फॉर द डाइजेस्टिव प्रॉब्लम. एक्चुअली मैम दिस प्लांट इज मेनली वर्क फॉर द डाइजेस्टिव इश्यूज और गैस्ट्रोइंटेस्टाइनल. प्लम्बैगो एज ए इंग्रेडिएंट यूज हो रहा है ना? उसके साथ और भी बहुत सारी यूज हो रही होंगी. यस, लाइक इट इज अ पॉली, इट मे बी पॉलीहर्बल, बट आई एम मेंशनिंग जस्ट फॉर प्लम्बैगो. तो ये you did not explain the MIC, am I right? Okay, कोई बात नहीं, आप देखना उसको. Thank you so much Aditi. Thank you for the suggestion also.
Thank you, Aditi. Thank you so much, Aditi, for giving very insightful information in your presentation. Now, we are moving towards our international panel discussion. And I request Mr. Piyush to please put the Zoom meeting on the screen. Hello. A very good day to all of our international panelists. I just want to confirm that my audio audible? Mr. Digambar sir, Dr. Sharad Vish, Dr. Vinitra sir. Yeah, you are audible. Okay, sir. Okay, as we progress with our conference, we are now moving to one of the most anticipated sessions, the panel discussion on BioStar 2.0 translational research for sustainable drug discovery and environmental health. We are privileged to have distinguished international panelists joining us online along with our esteemed members of the dais. The session promised to give insightful perspectives on bridging innovative research with practical application, fostering sustainable approaches in drug discovery and environmental health. Firstly, I should introduce all our international panelists. Dr. Balram Prasad Saha, Manager and HOD, R&D, Quest Pharmaceutical Private Limited, Adarsh Nagar, Birganj, Nepal. Dr. Mohammad Ali Mustafa, Associate Professor, Center for Health Research, Northern Border University, Arar, Saudi Arabia. Dr. C. Karthikeyan, Associate Staff Scientist, UMS College of Pharmacy of Medical Sciences, USA. Dr. C. Karthikeyan, Associate Staff Scientist, UMS College of Pharmacy, Medical Sciences, USA. Dr. Digambar Devekar, Root University of Cancer Research, Institute of Drug Discovery, Root University, USA. Dr. Sharad Vish, Assistant Professor, College of Pharmacy, Sihan University, Erbil, Kurdistan Region, Iraq. Dr. Marjan Dalil, Assistant Professor, Department of Pharmacology, Faculty of Pharmacy and Pharmaceutical Sciences, Tehran Medical Sciences, Islamic Azad University, Tehran, Iran. I, Ishan Agarwal, on behalf of SD College of Pharmacy, welcome everyone and one of our most prestigious alumnus, also Dr. Vinitra sir, Research Professor, Department of Pharmacy, Chungnam University, Seoul, South Korea. We extend a very warm welcome to all of you. So I start from Dr. Vinitra sir. Sir, we want your informative insights on this today's topic of the conference. Please, sir. Yes, sir, you are, yes, sir, audible.
First of all, everyone, it is my great pleasure and I will be here to talk about some of the friends who are in the Pharmacy, Professor Kumar. inviting me as an international analyst for this conference. And also I would like to thank all organizers, distinguished speakers and all participants in this wonderful opportunity. I am Dr. Vikas, so I would like to just a little bit lecture because as I had, they announced that AI was working at this purposes, so they just share my previous experiences. So I would like to share my experiences before start our conversation. So I am Dr. Vikas. I am currently working as assistant professor in Seoul University, South Korea. Previously, I worked in Yunnan University, South Korea, as an assistant professor. Then I started my postdoc here. After that, I started as a professor in Yunnan University. So currently, I am working assistant professor in Yunnan University. So I'm truly delighted to be part of the Bio Restart 2.0 Translation Research and Sustainable Discovery and Environmental Health. My research main focus on pharmaceutical sciences, AI-based drug discovery and translation research, which aim to laboratory discovery as well as connect to the practical clinical applications. So if I would like to more about to the link about this conference, and I would like to share about my experiences of to discover new things. So most of the researchers engage full-time active in the research. If anyone, the students are working as a master's and undergrad students, they know different kind of the research activity. As I learned many more kind of the educations, I gave my PhD, master's online. So I felt between some education and research because Indian education system mostly based like masters 1 year based on like some theoretical knowledge. But here in the Korea, from the first year, if you start the regulations like Master of Pharmacy, so you have to do like 9 hours research along with your credits. Credits, whatever subject you are learning. So here, student may engage different kind of research like cancer research, nanoparticle. Here nowadays, AI-based research are booming the platform to reduce the short time as well as provide the precision in drug discovery. So many research like big data analysis, if you familiar like Python-based data analysis, nowadays many hospitals, many industries, nowadays some laboratories, they are sharing. So maybe it may improve research as well as the skills of the students and knowledge of different people who are engaged in the scientific community throughout India. And we can give some fruitful outcomes within the short period of the time. So my point is that if we focus along with theoretical knowledge, like some practical knowledge with some courses like Master of Pharmacy, then people can learn more about the practical knowledge instead of the theoretical knowledge. So this is the most important thing nowadays. And if you know familiar nowadays, ChatGPT and many AI tools are more familiar to data research. There are many people raising some disadvantage about AI applications, but if we think that these AI tools suddenly transform the technology, so if we adopt these things and improve our skills in the research, then we can transform our ideas in the research, and we can accurately and more correctly we can define our aim and we can come in the conclusion. So we have to adopt whatever environment is changing nowadays. So we have to transform according to environment, what is going on. If I give the one example, most of the people in the universities in the South Korea, people are using AI tools openly. So many professors are allowed. You can use only ChatGPT or AI tools, paid version. So they are using, they are improving their skills. So the meaning of this kind of international instance, translation research, we use this technique in the India, many people have the languages. If they study from the Hindi board, so they come in the pharmacy, so they have different kind of languages. So they can improve their language proficiency using this kind of techniques, as well as they some professors, if they are developing their project funding, if they use this kind of ideas, so that they can improve their pharmacy for this kind of competition that they are doing, like they are businesses, they are taking as a complete some project, so they can implement this technique to build up some small project by using this technique. So we have to adopt this kind of things. So today the sustainability diversity is more important than ever because the world is facing challenges such as resistance, facing disease, environmental concern. So as I am working here in South Korea, so my basic research is going on some discovery, and I always implement these techniques to the development nanotechnology, some nanoparticles of this molecule, and deliver these approaches for the treatment like organic cancers. So my basic research is going on around this. So I am also around this one. I am guiding some master as well as PhD student here. So if we adopt only engage in the research, so we have to adopt latest some research articles. One thing I would like to say in this conference, here we have some exam, this kind of things in the master pharmacy and graduate. So some professors say to the student, you have to present some current research paper that one have the higher impact, like 10 to 15. Then those students who prepare their PPT for their examination, they can easily learn what is going on in the research. So if we a little bit change with our student, this kind of things, so they can easily adopt what is going on in the real world. So this is things, that is very important. Finally, I would like to say, most of the research that is going here, they are collaborated with many industries here, like pharma industries. If some project is conducted here, so they are collaborated some industry can fund in many laboratories with many professors and students and professors work on that project and finish that kind of the project. And so in this kind of the project, many students learn many things, new things, real practical things, and which is related direct application of and related to the clinical practice. So these kind of the things, if we implement in some colleges, some universities in India, and we can make our students more advanced and knowledgeable. So this kind of things are very important and very essential for our students. So we can knowledge as well as practical guidance to our professors, to our students, and the community. We can reduce the burden on different kind of diseases, nowadays. Diabetes, cancer are growing throughout the world, including India also. Many people are affected this kind of disease. So we are pharmacists, so we have to think related to the research and change our scenario, how we can this kind of issues, how we can solve this kind of issues. So we have to think around that. And then master pharmacy is very important. We have to strongly committed, and we have to attend international conference always, because I am here also. I always try to attend many international conference in the progress. So many students also attend. So they learn many conferences. Researchers in research in this area. So I wish to suggest to our students and faculties to attend some conference so we can get many things. So finally, I would close my talk. I would like to thanks all the participants and I wish the conference great success. I praise everyone who contributing conference science center. Thank you very much. Thank you.
Sharing so much insightful and meaningful times. I request to please join me in the next. I request Dr. C. Karthikeyan, Associate Staff Scientist, University College of Pharmacy and Medical Sciences, USA. Please, sir. Hand over to you.
Yeah, thank you. Good evening to you all. At the outset, let me thank the organizers of the State College of Pharmacy for providing me an opportunity to share my thoughts on a very important topic. This is a very timely topic, BioSAR 2.0, translational research for sustainable drug discovery and environment health. Now, why I am saying this? Because we are at the crossroads. And why is it so? Because AI, artificial intelligence, is disrupting all the applications as well. And it has disrupted the drug discovery research too. Because the genomic era, along with the genomic era, came a big data. Big data means large amount of data from multi-omic technologies. And they have been leveraged in drug discovery to come up with new targets, new class of drugs, and even immunotherapy. Now, since there is a large amount of data, it was not possible for us to handle it manually. So, although people have applied different statistical technologies, different programs to analyze this data to come up with rationalized targets, that has been time consuming, very expensive, and also a difficult task for a human to do. Now come artificial intelligence. Now artificial intelligence, because of the advancement in the computing power, it can leverage this big data to come up with more rationalized and novel class of therapeutics. Now, this is the crossroad, which means that the large pharmaceutical companies or multinational pharmaceutical companies are already adopting this technology, and several of the programs which are available all over the world have been providing services to these large pharma companies towards development of artificial intelligence technologies in drug discovery, and it has increased the pace, it has quickened the discovery of drugs. And this will become a challenge to our country because we lack innovation. Because if we don't adapt to this changing paradigm, we will lose out in this race. And it's very important for us to have dialogues or discussions on this topic, how AI can impact translational research. So that's why I have told you that this conference is organized at the right time on a very important topic. Okay, so how can we ourselves adapt? Because artificial intelligence is an evolving topic, which means that not much is known. Although you can put anything in your curriculum, because about artificial intelligence, but nobody knows how to apply. Because even pharmaceutical companies are learning how to use this technique in different. Because you are using ChatGPT, you may use Grok. Okay, but applications, depending upon the applications and upon the user differs. There is no uniform, there is no universal formula for applying this technology. So each of us are coming with our own innovative methodology to apply this AI tools in our day-to-day life. So it has been true for cancer, exponential drug discovery too. How one could leverage this technology towards drug discovery, whether is it for target identification, or is it for identifying a suitable subpopulation for a certain class of drugs, or is it to design new antibody drug conjugates, or is it to how it can be applied to design new vaccines, how it can be leveraged for evolving our immunology so many things can be done with the help of AI at a very rapid pace and less expensive, and you don't require a large amount of human resources. And for a market like ours, for a pharma sector like ours, which deals on generic-driven markets and which deals on providing cheap medicines to the world, this will be a changing moment because the pharma sector is moving away from small molecules towards large molecules, which means that antibodies, vaccines, and even antibody drug conjugates. So how can we adapt to this changing scenario or evolving scenario? We have to be prepared ourselves first, and it should start at the grassroots levels. It should start with the students like yours, being attending conferences like this. And your knowledge is as good as mine, and my knowledge is as good as yours, because you can use open source tools to learn about advanced artificial intelligence algorithms. How can they be used for your day-to-day problems? Because if I am applying AI in my research, for example, I got a proteomics data for one of the compound yesterday, and how am I using AI to delineate the data and to come up with a hypothesis and to come up with some kind of art to interpret the data? That depends upon me, and it will be a learning process. It will be a latent trial process. It will be an evolving process. So in this way, once you start to apply certain AI, artificial intelligence related tools in your research or even in your education, you will find that it is a very wonderful and a very useful tool. And along the process, you learn how to leverage this computing or this pace and this rapidity in your research or in your education. So that's why it's important for us to learn about these different AI tools and then to leverage them in your research or in your education. And other than that, it also gives you a better perspective on whatever, because it's an evolving field and it's a continuous process, because nothing has been optimized so far. So it gives you that competitive edge so that you can employ any of these tools and then come up with novel solution which doesn't exist or which is not possible because of our limitations. Limitations in terms of computing power, limitation in terms of resources. Because if you are using, for example, Perplexity AI, which was developed by one of our patriots, Indian. So Perplexity can do a literature search on any topic within 5 minutes. Okay, and it can organize the data for you. Even if you could leverage it, it can make a review and it can summarize all the research related to your topic. I use Perplexity every day in my research. So how to use these tools to come up with novel solutions for an existing problem, or how to come up with the innovative solutions, or how to come up with some kind of technology which has IP potential, then that can provide you the advantage of using this tool to advance your science or your education or even your professional life. So that's why these tools are very valuable, especially for a student. You can, for example, many of you would be doing your research projects in your final year. Why not use a tool, AI tool, to come up to frame a problem, to frame a hypothesis, or to come up with an innovative solution? And why not your research and then use that tool to come up with your own solution. So this is how we could leverage this emerging field, or we could use this emerging field to our advantage. So that's exactly why conversations like this or discussion like this should happen. And what can be done? Because right now so many service providers, they are using this tools to provide a workflow. For example, molecular docking. Okay, molecular docking because certain free algorithms or free softwares require programming knowledge. But there are now several workflows available on the internet which uses AI tool to provide a workflow in a very user-friendly platform. So similarly, you can also provide a workflow. It can be in any field. It can be retail, it can be manufacturing, it can be marketing, any workflow which can be used towards a certain application of benefit could be easily done using the AI fields. And that's exactly, students should have a thorough knowledge of all the AI tools available and at least have a limited knowledge on the applications of these AI tools. And then unless you use it, you will not be familiar with their applications or familiar with their advantages. And even right now, all these tools are available for free. Soon it will be available only on payment. So now is the right time to use all these tools to your advantage and to learn. Because as it is an interactive environment, it's not like, for example, you interact with an AI tool like you interact with a teacher. You give some and you gain some knowledge. And then it learns. It is a learning algorithm. So it learns from you and you learn from it. So it's a two-way process, and such an interactive process could enhance the knowledge of the student and enhance the knowledge of the student on a specific topic and keep the focus towards a problem and then come up with a solution. So that's how you can use these AI tools in your day-to-day life, in your education, and also in your professional life. So with these thoughts, we thank again the organizers for organizing such a wonderful conference, and I am hopeful that all these students who have participated and all the delegates who have participated in the conference have learned a thing or two about the new age translational research, and they will be using it in their research and education. Thank you.
Thank you, Karthikeyan sir. Thank you for your valuable time you have spared from USA. I know it's night over there. Thank you so much, sir. Next, I request Dr. Digambar K. Baker, sir, EDF Pune University, Institute of Cancer Research, Institute of Drug Discovery, Pune University, USA. Dr. Digambar K. Baker, sir.
Hello.
Over to you.
Yeah, good evening to everyone. I just need 5 more minutes to just start my laptop. I have joined through my mobile, so just give me 5 minutes. I'll be
That is a fine.
someone else to join, please.
Okay, sir. I request Dr. Mohammad Ali Mustafa, sir, Associate Professor, Center for Health Research, Northern Global University, Arar, Saudi Arabia. Welcome, sir. Over to you, sir.
Thank you to the organizing team for inviting me as a panelist speaker for this conference, Master 2.0 Translational Research for Sustainable Drug Discovery. So first of all, I would like to tell you about myself. I am Dr. Mohammad Ali Mustafa, the Associate Professor in the Faculty of Pharmacy, Northern Border University. in the Center for Health Research, Northern University, so here we are. So my research is focused on the nanotechnology, viability enhancement. So I am working on also the cancer research. So in my university we have now developed all this Center for Health Research where we do the translational research. Because most of the research nowadays now in the university, they are basic research. And when we see there are so many papers, but we see there are very less products in the market. So now my university think about that translation from the lab to the product. So that's why we have pharmacy, the medicine college, faculty member, and from the science college. All they work together and we are developing for the innovative product. There is a patent cells also there. So we are working on that. So, and we have a very good collaboration with different universities in India, like Chapman, like Bombay College of Pharmacy, IIT College of Pharmacy, Muzaffarnagar, for this research activity. So, like this, I am thankful for the invitation. Okay, thank you so much.
Thank you so much, sir. Dear sir, I request you. Okay. Thank you so much, sir. That you spared your valuable time. Now I request Dr. Sharad Vishd, Assistant Professor, College of Pharmacy, Sihan University, Erbil, Kurdistan region, Iran. Dr. Sharad Vishd, sir. Yes, sir.
First, thanks to the Institute of Pharmacy for the students to organize this international conference and for this invitation. I was with the respected distinguished chair, respected colleagues and other classmates. It's my privilege to share my thoughts in this international conference. I have started translation research for sustainable drug discovery and environmental. Basically, I'm working in Kurdistan region, Iraq. Previously, I worked in 6 different institutes in India and in the US. I have some around 5 bracket papers out of 9. I have some publications also, some good chapters, and also I deliver my lecture in different national and international conferences. Basically, here because of Kurdistan, it is not so much developed in pharmacy, but we are trying to develop more and more. Basically, we work on nanotechnology development. For example, I have one patent by which we can prepare the herbal nanoparticles without using any solvents. So basically, this is solvent-free method. Still, we are working on 2 more patents as it is a process patent to develop a new technique without using any solvent, how we can prepare the nanoparticle in a cheap way. The technology that I developed, it is around 7 to 8 times much cheaper as compared to the available methods. For example, if we use the supercritical fluid extraction, in that case also we can get the nanoparticles, but the equipment cost and spacing is too high. If we use the ball mills, it is also too high. But with my technique, it is not cheaper. It is like 6 to 7 times cheaper than the we are basically focusing here on here the Kurdistan region is not so much the national sources. So we generally organize trips and we collect different herbals and try to explore or get to know about the potential benefit or medicinal benefit of the herbs. Even we 1 year back, we discovered a new species of algae, but still the work is going on about this. And basically, we focus also, we have our own bachelor department, bachelor students here. So this time we are guiding only the bachelor students. But along with that, we also guide the master and the PhD students from the outside. Like, for example, students from India, IIM, or even here in some other university, IIM, master students also. And basically, as per the need of the current days, it is very important to collaborate with the different universities. For example, every university do not have sufficient number of equipment or we can or something. In that case, if we work in a collaboration way, it is easier to make for the betterment of the community and also to achieve the sustainable goals. There are 17 goals in SDG. So these goals also with this like collaboration. So basically, this is all about it.
Hello. Yes, Dr. Shansi.
Yeah, I introduce myself and what I'm doing.
Sir, your voice is not clear. Pardon, sir?
Yeah, I'm not.
Yes, sir, you are audible now.
Okay, previously I was not audible.
You were audible in most of the sessions, only the last line or 2 lines to me.
Okay, so as per I discussed in the last that we are working on the drug and polymer development. If there are 200 drugs, we try to make the esters. For example, the synthesis of curcumin and glyceric acid joining together and make a new molecule. So in this case, it shows a different physical properties and it will show the pH-dependent solubility and properties. So this is the one goal with the drugs. Second, we also focus on the polymer development. The polystyrene region is not so much explored. So we usually plan different visits in the forest, then gather the hubble, then work on that to develop or establish herb as a natural soil for the excipients. Even we try to explore the new bacterial species. We found one new algae here, which is not available in India actually. Wherever there is a, you can say, high source of sulfur water, it only grows on that side. It is generally white algae. It is not a green algae. And also here we are, because we are teaching only the graduate students, so we have only 4 years right now. Here pharmacy have 5-year course. So next semester we will start the final project for the students. But before that, even we have motivated some of the students, and they are doing research and collaboration with different universities, with the local region and also with the international universities. Basically, nowadays, every university do not have sufficient equipment. That's why it is very important to collaborate with the different international and national universities. And also this will help, because it's a community, to meet the goals of SDG, that is Sustainable Development Goals. There are 17 goals that are achieved in the SDG programs. With my goal of the 18, I achieved the SDG goal 3, 10 and 15. So basically, like our work nowadays, we are more focusing on SDG also and how we can meet these goals. Like this, we are drafting our research and projects. And again, I would say just thanks to all organizing team, delegates, and academic staff, and students to join this international conference.
Thank you, Dr. Sharad. Request you to turn off your microphone. Okay, thank you so much, Dr. Sharad, sir, that you have spared your valuable time and your concept of SDGs and the collaboration is really very important nowadays. Next, I request Dr. C. Karthikeyan, sir, Associate Staff Scientist, UMS College of Pharmacy, Medical and Medical Sciences, USA. Karthikeyan, sir. Sorry, sir, I think I have repeated your name. My mistake, my bad. I request Dr. Digambar Kevekar, sir, EDM from Institute of Cancer Research, Institute of Drug Discovery, Broad University, USA. Dr. Digambar, sir.
Yes.
Welcome, sir.
Am I audible?
Yes, sir, you are.
Thank you so much. I really would like to thank the organization of the International Conference on Biostep 2.0 Translation Research for Sustainable Drug Discovery and Development. Thank you so much for inviting me as a panelist for this discussion. So basically, I'm a postdoc research associate in Purdue University. It's not whatever you are going to pronounce it, but that was wrong. I'm sorry. It's a Purdue University. So, and I have heard like different panelists, applicants, and sir. So, I just want to share some of the work that we are doing here in Purdue. So basically, my work is on the microRNA. Very oligonucleotide therapies are very popular nowadays. It's taking attention from a lot of industries as well as academia. So we are trying to develop one of the microRNA, which is microRNA 34A, which is lost in most of the cancers. So we are trying to develop a therapy where we can deliver this microRNA to the cancer tissue so that it can inhibit multiple genes or multiple genes basically which are involved in the cancer, and that helps those genes help cancer to progress and make it more metastatic and ultimately leads to the that is ultimately failure of the cancer progress. But now we are trying to develop a therapy by which we can that we can use to target multiple genes, and how we are doing the advantage of different receptors which are expressed on the cancer cells. So I just want to mention the quote from my professor. And I give my condolences to my professor, Dr. Lau, who was a scientist at Purdue University, and he has almost 7 startup companies. So his work in 1976 in the Purdue University, and he was really interested in how we can take the advantage of these overexpressed receptor on the cancer cells. So he always used to teach us whenever we had one-on-one meeting or group meetings. So he always used to encourage us how if you are, for example, if you are reading an article, so how do we see that article? And when you read the complete article, you have to think how can I use this information, just not to apply the skills that I have, but how can I use this information and how I can use this information to solve, how I can use this information to solve a really challenging problem, or this information to solve something which I can do. And I had to think about it like in a normal way that this can be done easily. So that was his idea, and he was committed with his idea, and he developed a lot of technologies, and which ultimately got FDA approval. So just wanted to tell you one of the example of this work. He knew that the foreign receptors are overexpressed on the cancer cells. So then he started thinking how I can use this information, how I can leverage this information to develop some technology to deliver the payload to the cancer cell. So initially he started and he conjugated his folate with the dye. So dye was a fluorescent dye, and then he attached that folate to that dye, which is near infrared dye, and he developed one conjugate folate receptor with the dye. And he developed the technology, and he ultimately delivered that conjugate, the conjugate basically folate receptor and dye, and he administered that in the mice initially where the tumor was there. So, and he observed that all the dye in the conjugate receptor is just trapped in the cancer tissue and not elsewhere. So that's how he started thinking, oh, this is working, and then we can leverage this overexpressed folate receptor on the cancer tissue. Then he started commercializing, he started he asked certain companies to collaborate, and then that's how they started working on that same satellites dye, which got FDA approved in 2021. Yeah, so that's how you can think about something, and you can apply your knowledge, and you can really solve a challenging problem. So nowadays that dye is used in various countries to identify the cancer tissue, whether it is metastasized or particular tissue specific. And surgeon can administer the dye to the patient, and they can resection or cut the tumor cells from that a particular tissue. So that's how it is helping the patient basically throughout the world. Another example I just want to share. That another cancer type is there, which is prostate cancer. So again, he started thinking how I can treat the prostate cancer, which is very common problem in the main population in the world, like not in the United States, but it's everywhere. And it is the second leading cause of the death. Prostate cancer is the second leading cause of the death in main population. So now he started thinking about that, the prostate cancer. Then he, again, as I mentioned, folate receptors are overexpressed on various cancers. Though it is already, though it is like normal, this expression of the normal tissue is very less. But he started to think about the prostate cancer. So he found out that there is another type of receptor overexpressed on the prostate cancer, which is prostate-specific membrane antigen. So then he took the advantage of those overexpressed receptors on the prostate cancer, and then he arranged one radioisotope, which is lutetium-177, and he created that lutetium-177 with the DOTA molecule and attached the DOTA molecule to the PSMA binding ligand. PSMA binding ligand has high affinity for the PSMA receptor overexpressed on the prostate cancer. And then he delivered that conjugate, lutetium conjugate to the animal models initially, and he was surprised that it goes into the prostate tumor, and if that conjugate remains in the prostate tissue. So what's going to do is, because lutetium-177 is there, so it's going to emit the radiations, but that radiation is going to be localized to the tumor tissue. In the tumor cells in the closer proximity, not like other parts of the body. So that's how that lutetium-177 radiation kills the cancer cells. So see how he has conceptualized everything, how I can deliver this nutrition once around this hand so that it won't harm the normal cells in the body, but it will only kill the cancer cell in the tissue specifically. So that's how we developed another technology which is known as PHARMACY, which is approved by the FDA, and then PHARMACY was the technology developed by the Philip Lab and the Purdue University, and ultimately that technology was sold to the big pharma company, and that's how it is working for the patient. And there are so many technologies are in the pipeline. I cannot disclose those things, but 7 to 8 of these molecules are in the clinical trial phase 2 and phase 3. And the different for the different diseases, for example, inflammation, for viruses, viral infection. There are a lot of things working in the different directions. So I'm just giving you an example. But right now we are working in another aspect of the disease cancer. So we are trying to develop a technology where we can, deliver oligonucleotides to the cancer tissue so that it is going to remain in the tissue for like weeks. So if you have heard about the GAMNET, which is oligonucleotide therapy approved by the FDA for the liver treatment. So the oligonucleotide remain in the tissue for longer period. So you can, if you are delivering something which can remain in the tissue for longer period of time, so that's how you can reduce the frequency of the dosing, and you can make the patient comfortable with the technology or the doses comfortable with body. But so as I mentioned, microRNAs or the antisense oligonucleotides, you cannot deliver them as such because they will be dispersed like in the body throughout. So you want to deliver them like specifically to the tissue where you want to deliver them. In our case, we deliver the oligonucleotides to the cancer tissue. So we are attaching those oligonucleotides with the different ligands which has high affinity for the receptor overexpressed on the cancer tissue. So, and oligonucleotides as such you cannot deliver, like for example microRNA, they are very sensitive. Once you will just inject them in the body, they will immediately destroy. Why? Because they are very sensitive to the RNAs present in the body, in the serum. So what we are doing, we are making some modifications in the RNA backbone, and then we are testing them whether they have same functional property after modifications or not. So what we are doing 2 modifications in the sugar backbone. Sugar portion we are replacing 2-hydroxyl group with the methoxy or the fluoro because those groups are very sensitive. They participate in the hydrolysis, and so we are replacing those hydroxyl group and the fluoro group for the sugar, and we are putting some sorry. So we are removing those hydroxyl group and putting O-methoxy or fluoro, not other modifications, and we are also replacing phosphodiester backbone with the phosphorothioate backbone so that the enzyme RNases won't recognize those phosphodiesterases and won't leave the RNA backbone. So that's how we are making more stable microRNA sequences or oligonucleotides, antisense oligonucleotides as well. Because my friends, my colleagues are working on that direction as well. So that's how. So, just a small things we can improve the quality, or improve the overall life of the conjugate. So that's how we are making. When we tested the modifications, we saw that it is stable life for in the blood serum. When we tested in the blood serum, we found out that it is stable for 48 hours without any degradation. But when we compare that with the normal without modifications, sequence is not at all stable in the blood serum. In 5 minutes, it started degrading. So that's how we can improve the stability basically of the microbiome. And we are also working on the breast cancer, the prostate cancer, ovarian cancer, and the bone cancer as well. So, and as Dr. Karthik has mentioned here about the AI tools. So we are also using some AI tools such as AlphaFold to see the structure of the RNA. We are also using the Schrodinger software to see the document interaction, all this the dynamics. One molecule to the protein backbone. In Purdue also, one of the Professor Dyson Kiana, he's developing another AI tool where we are training our model to for the RNA interaction basically. So that model is for prediction of the RNA structures and the short RNA structures and its interaction with the genes of the proteins. So how we can so that's how we train the model, and then we get the information back from the models. So, and we are also using like other online tools to predict the structure of the RNA and distance of the nucleotides or the siRNA basically. So the overall, I just want summarize now. So whenever the students, basically the masters or the PhD students, whenever you just go through any articles, read it carefully and don't read all the unnecessary information, but whatever the project that you are working on, just read it, the article as article mentioned perplexity. It's a really helpful tool for the students. And you can use that tool. I use that tool a lot. I use ChatGPT as well to get information. What is like? What are the? You can ask anything to those AI tools, and they're gonna give you all the information related to your specific the work. So I just want to summarize. Whenever you read any paper or whenever you use any tool, just see. Just try to get as much as information related to your work and how. Just think about how you can use that information to improve the quality of your work, to improve the quality, the lifestyle of the patient. How can you help the patient which are really in need? And which are those who are suffering from particular disease. Any disease, you can start working on any disease, and just you have to think how you can use those information to make the patient's life better in all the hospitals. Thank you so much. I would like to thank the organizers for giving me this opportunity to interact with this audience. Thank you so much.
Thank you, Digambar sir. I request you to put your mic off. Okay. Digambar sir, really, it was a very informative session that you gave, and a very wide use of research articles and the AI tools in modern drug discovery, perplexity and ChatGPT. We all use all those tools. And thank you so much, Digambar sir. You have spared your valuable time from all the way from USA. And we, on the behalf of SD College of Pharmacy, thanks all the international panelists which have spared your valuable time. Thank you so much, sir. Thank you to all.
I hope that you all got
speakers. And now I request our organizing secretary of the Student International Conference sponsored by UPCST, Dr. Bhupen Singh sir, on the rise please, on the stage please, for the concluding report of today's session. Dr. Bhupen Singh sir.
Thank you, sir. The first day of CST-UP sponsored 2 days International Conference Biostat 2.0 Translational Research for Sustainable Drug Discovery and Environmental Health, organized by the SD College of Pharmacy and Vocational Studies Muzaffarnagar, concluded successfully with enthusiastic participation of researchers, academicians, and students from various institutions. The conference witnessed a remarkable academic response with a total of 185 research presentations registered, including both oral and poster presentations, out of which 92 presentations were scheduled and successfully conducted on day 1, providing a vibrant platform for researchers to present their academic findings and scientific ideas related to the discovery of drug, environmental health, and translational research. The scientific session was enriched by the insightful expert lecture delivered by the distinguished speakers. Dr. Harish Dureja sir delivered an informative lecture highlighting the importance of advancement and research perspectives in the pharmaceutical fields. The session was highly appreciated by the participants for its scientific depth and practical insights. Another enlightening lecture was delivered by Dr. Dinesh Kumar sir, who has shared valuable knowledge and research experiences, inspiring young researchers and scholars to explore innovative approaches in drug discovery and environmental sustainability. Throughout the day, multiple technical sessions for oral and poster presentations were conducted smoothly under the supervision of session chairs and organizing committee members. The session encouraged active interaction, constructive discussion, and knowledge exchange among participants. We have successfully coordinated the international panel discussion with our panelists for US, South Korea, Iraq, Saudi Arabia, and Nepal. The conference will continue on day 2 with further keynote lectures, technical sessions, and academic discussions aiming to promote collaborative research and innovation in the field of pharmaceutical sciences and environmental health. Thank you very much.
Thank you so much, sir. And now I request all the participants that today's session is closed and आप सब IT का प्रबंध है उसको लेके जाइएगा and कल का रिपोर्टिंग टाइम 10 AM रहेगा तो और वैलिडिटी सेशंस भी कल रहेंगे और हमारे और भी स्पीकर्स हैं जो ब्रोशर में मेंशन है वो भी कल रहेंगे तो सभी स्टूडेंट्स सभी पार्टिसिपेंट्स से रिक्वेस्ट है कि आप लोग टाइम से आइए ताकि आप कोई भी इंफॉर्मेटिव सेशन मिस ना करें थैंक यू
बंद करो और अपलोड करो 3 घंटे
मैम देख लो चश्मा